Morning or good afternoon, depending where you are in the world. I'm Diane Joseph McCarthy from Boston University, and I'm a member of the organizing committee. And it is my pleasure to welcome you to the AI for Biomedical Research and Drug Discovery Symposium of the AI Alliance. So next slide. So as I think you can see, we have an exciting lineup in store for you for the next few hours. There will be two keynote sessions with a short break in between. And during these sessions, we'll hear from global leaders in the area of AI and the life sciences and drug discovery. These will include speakers from several European countries, as well as the US, and representatives from academia, from government, and from industry. Then there'll be a session on demos and walkthroughs of open source tools and models and AI agents for biomedical research with presentations by the developers of these technologies, and I think from BU and many from IBM. And finally, there'll be a panel on collaborative benchmarking for biomedical foundation models. And that will undoubtedly prompt a lively discussion with panelists from Hebrew University, OpenADMET, and the Structural Genomics Consortium. And finally, we'll conclude with a few closing remarks. So next slide. Okay, so the vision for the AI for Drug Discovery Working Group is to increase efforts in AI for the life sciences in order to drive drug discovery, and to do so responsibly through open collaboration. And the mission is really to establish a world-class open research community to drive the development, evaluation, and large-scale adoption of AI technologies. And this AI Alliance Working Group was started in October of 2024 at the AI for Drug Discovery Open Innovation Forum that took place at Boston University. It was a very successful event with over 160 participants, and you can check out the website, which is given here, as well as you can listen to recordings of the presentations. In addition to insightful speakers and panelists, it also highlights paths to scalable development and deployment through showcasing two open source models through the IBM OpenAG framework on Red Hat OpenShift AI platform on Mass Open Cloud. Next slide. And the AI for Drug Discovery Workshop has several ongoing initiatives. The National AI Research Resource Pilot, which you can find out more information about by following the link there, is designed to facilitate collaborations and share resources. There are two projects that the group is actively working on. One involves benchmarking and is participating in the DREAM Challenge, where the challenge was to predict the binding of molecules to the WDR91 target and to demonstrate the value of fine-tuned foundation models versus pre-trained foundation models and non-foundation models. And it's now in this stage on the right side of the graphic of experimental testing. And the plan is to participate in another challenge following that. The second project involves AI-driven multimodal approaches to decoding cancer, where the goal is to examine the successes and limitations of foundation models in studying cancer from the cellular and microscopic perspectives. The team is using large language models to automatically analyze scientific publications and extract relevant facets of interest. And so they've already identified 40,000 scientific publications on AI, cancer, and omics, and have devised various facets to include that are related to the hallmarks of cancer and its various types. And so the next steps there will be to do large language model assisted facet extraction and analysis and interpretation of the results. So I think there's really a lot of exciting work going on as part of this working group, and hopefully that will continue and expand as we move forward. We cannot hear you. You're muted.
Oh, apologies. Yeah. So hopefully you can hear me now. So I'm just introducing, thank you, Diane, just introducing the first speaker that we have today. So first speaker, so it's a very busy and exciting agenda. So we please ask all speakers to make sure to stick to the 15 minutes for their talk and up to 5 minutes for questions. I will mention something at the mark of 15 minutes or so. So please, for all participants, post your questions on the chat, also via the Slack channel as well. We invite the discussion during the chat in the chat room, but also in the Slack channel. So without further ado, I'm just going to introduce Professor Schuler-Furman from the Department of Microbiology and Molecular Genetics at the Institute of Biomedical Research, Faculty of Medicine, Hebrew University of Jerusalem. So today the keynote is about AI in protein modeling and design, insights from peptide to protein applications. And Ora, I've made you a speaker and presenter, so hopefully you'll be able to share your screen and presentation. So, Ora, are you able to share? Just see. I think we do have Ora in the room. Just want to make sure.
Okay, I need to. Hello.
Hello, we can hear you now. Yes. Hello.
I just have to find my window that I want to share, and then I can start.
That's great. Thank you.
Okay, can you see my screen?
Not yet. But now we can. I think now it's loading. Yeah, we can. Thank you.
Yes, we can.
Yeah, because I can see. Can you see my screen? A hot score picture? Okay. So hello. Yeah. Thanks for inviting me for presenting some insights about what I've learned and memorized with the AI evolution. Okay, so I'm going to talk about AI modeling of proteins and design, and specifically relate to what we can learn from peptide protein applications, both on what AI has learned and memorized, and both on what we can actually do with the new tools. So I want first to talk about the AlphaFold revolution. So last year, the Nobel Prize went to two different groups that have worked on protein structure prediction and design. The actors are David Baker from the University of Washington, which developed de novo designed proteins for the first time and has funneled the advances for protein design, but also structure predictions that I will show in a moment. And the second part went to AlphaFold, which was developed by Google DeepMind, which I assume that you all have heard about. These are two different directions that converge, and I will try to provide the basics that allow these two directions to develop. So what we can do today, we can predict structures of proteins at an unprecedented scale and accuracy. Here I can show you a few very, very impressive examples. This is, for example, this is a very, very large, this is a nuclear pore that basically allows proteins and other features to enter and exit the nucleus. It's hundreds of proteins that are put together, and we can today model these structures with AlphaFold. There are other enzymes that we can model, for example, here, where we can learn what would be the way to actually inhibit them or modify them. And on the other side, we have the protein designs, which are not less impressive, where we can today generate nanomaterials that of very, very large size and increasing complexity. We can today generate motors, and you say it, you name it, and it's going to work. So this is a very, very exciting time. What made this possible? So there are three basic tenets that allowed this to happen. The first is basic research. So there has been about 70 or more years of protein research that has allowed us to understand what actually proteins are and what defines their function and their structure. So one of the basic assumptions that we have been able to formulate is that we can define the problem of protein structure definition or prediction, and also design as an optimization problem, which involves sampling a very large number of different possible conformations and ranking them based on energy function. And this is based on simulation of what we learned about biophysics, how actually nature works. That proteins that want to be stable, they need to have a very low energy. So according to the Boltzmann principle, they will remain in the same conformation. And in order to accelerate this, there have been other features that have been necessary for this. And the second tenet is the big data part. So we need a lot of data to actually understand and learn about these systems. And part of it is, for example, the very large number of solved structures that have been generated by very, very intensive work of many courageous biochemists and structural biologists, but also the increasing information about genome sequencing, which I will talk about in a moment. And the third part, of course, is deep learning that has allowed us to actually combine these two different tenets to devise models that are very, very accurate using new architectures and optimizations. So the first part was a breakthrough that was made possible actually by the Baker Lab, which was called Rosetta ab initio modeling, which was the first time where we were able to solve the structure, to predict the structure of a protein just based on ab initio modeling without relating on other solved structures. And the way this was done is basically by a very efficient way of reducing the sampling size that we need to sample to find this correct conformation using fragments. The second part was made possible by the amass of many different sequences, which allowed to analyze coevolution in along evolution. So this means we can identify positions in a protein that change together. When they change together, we can assume that they actually change together because they are not close in space. And this basically allows us to understand that the protein structure is basically a contact map. A contact map that is composed of residue pairs that actually show some correlated evolution. And these contact maps represent the structure. The more contacts we can predict, the more accurate is this structure defined. And people very quickly recognize that this is actually an image. And if you can use and because it's an image, we can actually use image recognition protocols and networks that have been developed in the same years and deep learning to actually identify those images. So like here, when we see a picture of David Jones, one of the main developers of these neural nets at the beginning, you can basically define an image very well if you define its contours, because that already defines what types of features this image includes. For example, here a person. You can do something similar with these correlation matrices, just find the contours or the critical points that are needed to actually understand what the proteins look like. So this is the second advance. And when we look at AlphaFold, this is the first time where this was extended to an end-to-end architecture. So this is the architecture of AlphaFold. Again, we have this sequence. We can use multiple sequence alignments to generate these pair representations where basically we find correlations between positions. And there is some sophisticated network that at the end gives us these contact maps. But in addition to these contact maps, we also get the final structure, which is this end-to-end architecture. And this has really led to a very, very big revolution that has changed our field completely. So in addition to providing structures, AlphaFold also provides some confidence, which we can use to understand where AlphaFold is sure about its predictions, where there might be some correct answer, and where there are regions where it cannot say a lot. So, and AlphaFold 3 that was developed, just came out, extends this to molecules beyond proteins. Okay, so AlphaFold gives us a lot of answers. It allows us to accurately model at large scale protein structures. We have structures now for practically any protein that you can imagine. It identifies unstructured regions, which is not as important because these unstructured regions have other features than structured regions, and they are really important for regulation, which I will talk about in a moment. It provides guides for biological research. Biologists can use these structures to actually understand what their proteins are doing. And there are a lot of other things. But there are also questions that arise. So what has AlphaFold learned? What did it learn? What did it memorize? If we go back to this Boltzmann principle and to this energy landscape, did it learn what an energy function is? or did it just memorize local features that it can reproduce and identify in structures? Second question, is it extendable? So we have now extensions to small molecules and so on, but where can we actually generalize AlphaFold beyond what we have today? Can it be adapted to interactions? Can we look at the dynamics? The fact is that the AlphaFold models tend to converge, so we can't really learn about protein dynamics based on these models, and there is a lot of work going on in that direction. And I want to talk today about how we can actually use protein-peptide interactions to find out more about these questions. So modeling protein-peptide interactions is a challenging task because of the fact that the peptide does not adopt a structure before it actually binds. And there are many ways of how proteins bind with their peptides. This is a very efficient way to achieve high affinity interactions with very flexible and transient motifs. It's like when you communicate with SMS with other people, it's a short linear motif and not a short motif sequence, not short message service, it's short motif sequence. So these SMS are really dependent on the context. And based on the context of people that text together or proteins that interact together, we can actually change the way these interactions are formed and regulated. So very flexible, very transient, and very context dependent. So these are very nice, interesting interactions that play many interesting roles in biological regulation, but they're also a very stringent test case for our ability to model things because of their transient and weak interactions. And it is challenging because these are very small interactions, so there is not a lot of enthalpy to be gained. And also, there is a lot of entropy to be lost because the peptide is flexible. So nature has found some solutions to adapt this, and we have used these solutions actually to develop protocols in our lab to predict these interactions at high resolution. And we started this with Rosetta FlexVector, which was an initial protocol to model these interactions, to refine this interaction at a high atomic level, high resolution, so we can actually use these for further drug development and assessment of interactions. But then the question came, how can we do this global peptide docking if we don't know where the structure, where the peptide binds? And we have developed different protocols during the years that basically are all based on basic concepts that we defined. And one of the concepts is that a protein that binds a peptide is basically complementing its full structure. It's like something that is missing the last bit to actually get a stable monomer. And based on these assumptions, we can produce different protocols that actually solve that problem very well. And of course, I won't go into details because I don't have time, but of course, AlphaFold solved us this problem because basically from the beginning, we could see that even though AlphaFold was presented as a monomer prediction, we can actually add this peptide as an additional addition connected by a flexible linker and predict the structure. So we can see here what we did. So if we add this artificial linker, we can see that AlphaFold was able to actually predict the structure of these peptide-protein interactions very, very well. And sometimes it failed, but we can see that it fails, and we understand why. So this was a very, very big advance for us, and also in general for the assessment whether AlphaFold has learned or memorized, because this assumes this basically suggests that even though AlphaFold was trained for monomer structure prediction, it can actually predict much more. This was one of the first examples that shows that AlphaFold is extensible. And we can show here in these pictures, these are plots where we have like some measure to the correct structure, which is 0 here, and some AlphaFold measure. And you can see that in many different cases, we can see that the ones with high values are the ones that are actually good predictions. And we are only few that we are missing and that we can further refine and develop new protocols today to actually solve most of those. But most of this has been solved. And so we applied this to a large set of motif classes that are collected in this N database. And we could show that also in these cases, we have a lot of predictions where we can actually get high confident models that are very, very good, very accurate. We can also, importantly, we can also predict the effect of the mutations. If we look at these interactions and we mutate the critical residues, we can see that the changes in the confidence are very significant. So in yellow are the ones if we do single mutations, in blue are the ones that we do double mutations with 2 different AlphaFold measures. You can see that we can actually predict the effect of mutations very well, which is not a solved problem in general. So that's a very encouraging result. I don't have time to talk about many other things, but I will talk about one application that demonstrates how the field has advanced that we have done in our lab, together in collaboration with the lab by Jakob Nielsen in Copenhagen. So we have been looking at a protein that is called PP2A, it's a phosphatase. Phosphatases remove phosphates from proteins. So if a protein is phosphorylated by a kinase, this phosphorylation can be removed by a phosphatase, which allows on-off switches and regulation post-translation. The important point about phosphatases is that they, by themselves, do not provide the specificity that is needed to actually identify the substrates that you want to unphosphorylate, the phosphatases. There are these additional units, which are these regulatory units here, that confer specificity. So this PP2A, based on whether it's bind to any of these three subunits, will bind and modify different substrates. And while there are hundreds of kinases, there are only tens of phosphatases, and they get their specificity by binding to different subunits and allowing for specific modifications. So while the lab by Jakob had identified for B56 a very nice binding motif of all the substrates, he couldn't find any for the 10 substrates that he had found for this PP2A. So we modeled these interactions with these different substrates. And what you can see is these are AlphaFold models. This is the B55 subunit, and you can see the different models. In blue are confident regions, in yellow are regions that are less confident. And we could immediately see that there are many different ways of how these substrates will bind, but all to the same pocket, but in a very different way. So there are these canonical alpha loop helix, alpha helix loop, alpha helix models. There are others that bind in a very different way, and we could actually validate these binding models by experimental validation. And then there is this other one that we don't really understand how it works. We actually do, but I don't have time to talk about this. If you want, you can send me a message and I will be happy to talk about this more. But what I want to focus on now is this type of interaction. We reasoned that because of the fact that we can actually model these interactions very well, and because of the fact that we had actually mutated them and they worked the way that we expected it, that mutations that were at the interface really abolished binding, we reasoned that we might be able actually to design a binder that will be much more stronger than what these binds, and define a binder that will basically inhibit this phosphatase. So we started with an helix loop helix model as a template, and we applied a protocol that is called Protein MPNN, which is a design protocol, a deep learning design protocol developed by the Baker Lab, that basically designs sequences that will allow this structure to bind very highly or strongly best to this protein. And the design sequences were refolded, so we actually used these design sequences and refolded them with AlphaFold. We predicted the structure of this P55 subunit together with the design. And then we selected those that were actually modeled to be to bind exactly in the way that we actually had predicted. And from those, we selected the top ones, and the group of Jakob Nielsen tested them. All of them expressed, except for the ones where we used a different protocol that we don't use anymore, obviously, because it doesn't work. And what was important here, it's not only that they worked, but the binding affinity is sub-nanomolar. So this is a very, very strong binding. And when we mutated the structure at critical positions, the binding was abolished. So this is something of unprecedented quality, where we have a one-shot prediction where we get a sub-nanomolar binding for the first prediction without any further experimental refinement. More importantly, this binder is very, very specific. If we use it and we pull down all the proteins that are available in these cells, we can see that it pulls down only this, the B55 and the PP2A structure, which means that it is very, very specific. And we can also see this with proteomics approaches. We can see that the proteins that are not bound anymore now to this subunit are the ones that are actually the substrates. And most importantly, when we look at the phenotype, you can see that adding this inhibitor elongates the G1 period in the cell cycle, which basically means that we can actually, with this binder, we can actually control the length of the cell cycle, and you can by yourself imagine how important this can be for drug design, the targeted drug design that will basically allow us to actually control cell cycle progression. So what next? We are looking in our lab at many hints of how we can actually interpret the results from deep learning. We are looking at hidden correlations to detect additional motifs that could be important for peptide protein binding. We are also interested in overconfident predictions. What do they reveal? AlphaFold sometimes predicts with high confidence structures that don't make any sense. And the question is, why does it predict them to be so with such high confidence? It could be that that it's simply wrong, but it could also be that there are some secrets that it knows that we don't know, and we have a number of examples where we can actually show that AlphaFold has revealed new features that we couldn't have imagined before looking at those models and the confidence. We are looking at models, we are training models for functional peptides to try to understand what these peptides do. We are looking at orphan receptors to try to understand which receptors bind to which peptides, and for designing specific markers, you can imagine designing a peptide that will bind to a specific receptor on the cell and can basically serve as biomarker, or another peptide that will basically bind to some cancer target and inhibit progression of cancer. We have one example where we have shown in vitro that we can actually design a specific binder to one of those targets. So that's it. This is my lab, and I want to thank everyone. And I think I'm on time, right?
I think we're out of time. That was great. Right, perfectly on time now. Or thank you so much. Yeah, thank you. We had allocated about 5 minutes for questions, but I think we ran out of that time. So I was going to invite if there's one very, very quick question that we could ask now, or please ask your questions with more time in the chat, and we'll get to you as soon as we can, please. Okay, so maybe let's do that. Let's take the questions on the chat. So, Ora, please take a look at the chat as people might start reaching out with questions. Thank you. So, just going to hopefully share my screen now, and yeah. So next up is Professor Borgward from as the director of the Max Planck Institute of Biochemistry. He also leads cutting-edge research developing novel machine learning algorithms to detect patterns and statistical dependencies in large data sets from biology and medicine. So without further ado, Professor Borgward, please go ahead.
Thanks for having me. It's a pleasure to present my work at this meeting. I'll go to full screen mode. I hope you can see my slides now.
Yes, perfect. Thank you.
Wonderful. Okay, as I said, it's a pleasure to be here. I want to tell you about our efforts in machine learning for biomarker discovery. My lab, first at ETH Zurich and now at the Max Planck Institute of Biochemistry in Martinsried near Munich, has been working on this topic for at least 15 years. And we have seen the advantages and disadvantages of different approaches to biomarker discovery. And I want to summarize these findings over 15 years to you in these 15 minutes. So it's an ambitious plan, but I'll try to get there. So let me first motivate why we are interested in this problem, or why we hope that machine learning can play a role in biomarker discovery. So Ora mentioned this in her talk already very, very nicely. So there have been very big successes using machine learning over the last 10 to 15 years. So problems that were long deemed unsolvable in the decades before, like classifying images, recognizing speech, text generation, or predicting protein structures, all of these have been solved to a very large degree. In the meanwhile, the latter one even resulting in a Nobel Prize, as you have just seen in the previous talk. Of course, this huge success now triggers the question, can we solve other fundamental tasks in the sciences and in other domains using machine learning? And one that bioinformatics, biology, and the life sciences are in particular interested in is biomarker discovery, which I want to first define before diving into more detail. So biomarker discovery is of utmost importance in a subfield of life sciences, which is systems biology. And systems biology studies the interplay of the components of a biological system and the functions that these components give rise to. And we often refer to these as the phenotype of this system. And I mean, when I here speak about a biological system, I mean this in the broadest possible sense. So from the molecular level, from nucleotides making up a gene and its function, to amino acids giving rise to a protein and a particular function or structure of that protein, from cells forming a tissue, from organs forming a patient, and even further to individuals forming a population, which you then study in epidemiology. So all of these are, in my definition, instances of systems biology. And now the task to find out which components of a biological system are predictive for a phenotype of interest, this is exactly the search for so-called biomarkers. So the biological properties of a system that are associated with its function. Now the big question in my lab's research, but in a whole sub-branch of bioinformatics, has been for a number of years, can machine learning revolutionize biomarker discovery as well? And in order to understand the answer and to give the answer to this question, I want to summarize, first of all, classic biomarker discovery and what has been achieved there in general and in my lab over the last 15 years. The very classic biomarker discovery is rather old and has been popular for a long time. Fields like statistical genetics, for example, in so-called genome-wide association studies, perform biomarker discovery. And so this is no surprise. Discovering the genetic basis and the genetic properties that are associated with complex traits and complex phenotypes is one of the open unsolved problems in genetics, in computational biology, and in the life sciences in general. In fact, before the human genome, as you all may know, before the human genome was sequenced 25 years ago, there was the hope that afterwards you could associate every complex trait with one sequence position in the genome, so-called single nucleotide polymorphisms, or short SNPs, but that hope was not fulfilled. Even this turned out to be so much more complicated that even a term was coined for this failure in genetics, the missing heritability, so that even traits for which you knew, or where we know from twin studies or family trees that they are heritable, we could not predict them using genotypic information about individuals. So this was a big disappointment after the sequencing of the human genome. Now, what was the reason for that? A lot of reasons have been discussed. One potential reason may be the simplicity of the models that had been used. They were based on this idea that there might be one locus, one position in the genome that is associated with phenotypic variation. So to describe this here, what is happening in this toy example above, in a typical statistical genetic study, you would have a collection of individuals for which you have genotypic information, often these SNPs that I mentioned before. Typical number of these SNPs in today's studies is hundreds of thousands to millions per individual, and you would have at least one observable trait. Here, in this toy example, it's the height of the plant. And then you would, in a classic genome-wide association study, ask which position in the genome is most associated with phenotypic variation. And it turned, unfortunately, out that for the vast majority of complex traits, you can only explain very little phenotypic variance using this approach, using this so-called single locus approach. And this was clear around 2010. With very few exceptions like human height or lipid levels in the blood, the variance explained was at most around 20% for most other complex traits. Back in the days, I was already in the field of bioinformatics, and my lab and I wondered whether the solution to this could be found by making these models one step more complex, in that we go from single locus association mapping to two locus association mapping, also referred to as epistasis search in genetics, and to go then further from interacting pairs of SNPs to interacting higher order sets of SNPs. And for a number of years, we in fact developed algorithms, efficient algorithms for exactly this task, for finding groups of SNPs that are jointly associated with a phenotype of interest. And we published a series of papers under the name of significant pattern mining or combinatorial feature selection for this task. So up until very recently, so one of the latest, most recent contributions here is a ICDM paper from 2023 by Pellizzoni et al., where we explain how to compute the false discovery rate using these approaches. Now, they are, and I know that many of you have an algorithmic background, so these algorithms are by no means simple, because there's a huge computational and a huge statistical challenge involved. The huge computational challenge is that when you have 1 million SNPs, so 10 to the 6 SNPs, when you go to pairs, you have a search space of size order 10 to the 12, that you have to search for the SNP pairs that are most associated with a phenotype of interest. And this is not only a computational search problem, it's also a huge statistical problem, because every potential pair represents a hypothesis that you are testing. And whatever correction technique you use, like Bonferroni, or more sophisticated, like the false discovery rate, if you apply them to such huge numbers of tests, then they lose all statistical power. And how to overcome these two problems was a huge challenge, but in fact, under a certain number of conditions, I don't have the time to list them here now in much detail, but under a certain number of conditions, we could actually solve these problems thanks to the so-called Theron's trick from 1990. This was an old statistical trick that said that if you use discrete test statistics, for instance, Fisher's exact test, then not all hypotheses in this huge space of hypotheses that you are testing are testable. So testable means they can reach statistical significance at all. So Theron claimed and proved, in fact, mathematically, that many hypotheses cannot reach statistical significance in such a setting. And he also proved that untestable hypotheses can be ignored in multiple testing corrections, thereby bringing down the actual number of hypotheses that you have to correct for in multiple testing correction. This very interesting result was rediscovered by a group of Japanese scientists, Terada et al. In 2013. This was a PNAS paper. And back in the days when I read this paper, I realized that this actually applies to a lot of problems in computational biology, and that we might be able to solve this higher order interaction discovery problem using Theron's trick in a number of problems in bioinformatics. And that's what we did then for almost 10 years. So we developed, I give here only 2 examples, but we developed efficient algorithms for finding sets of neighboring genetic variants in the genome that are jointly associated with a phenotype of interest. And rather recently, we also did the same for a network of genes, where we are trying to find sets of neighboring genes in this network that jointly show a strong association with the phenotype. That said, there still remain a number of limitations. It's the aforementioned assumptions that we make in this approach are a limitation. For instance, Theron's trick holds if you have a data set of fixed size n. And the other limitation of this approach is that the phenotypic variance explained remains low to moderate. So while we could solve the algorithmic problem, it's not that we then discovered like pairs of SNPs that would explain way more than 50% of phenotypic variance in these genome-wide association studies. So that's where I end this part here of the talk. If you're interested in more, I've given the URL here. You don't have to remember it. if you search for significant pattern mining, this should be the top 1 hit. And then we have a whole list of all the works that we did on this topic. Now, of course, this didn't satisfy us fully, like that we couldn't explain more variants or that we couldn't predict phenotypes with higher accuracy. Of course, we also witnessed this huge revolution of deep learning and of big data. This was also very nicely mentioned in the previous talk. So in the 2010s, in the mid-2010s, I would claim, massive collections of sequence, time series, and image data became available in the life sciences, from plant breeding to bioengineering to medical informatics. And my lab started building models for predicting phenotypes for these different domains. In fact, all of the domains I just mentioned, but in particular, medical informatics. And I just want to mention here a few things that we did over the last few years. So one area of medicine that we found to be very interesting is intensive care, because in intensive care, you collect a lot of information about a given patient, like the health history, you have the treatment information, you have these vital signs that are being measured by sensors, you know all of this about a patient. And then a very clinically relevant problem is to ask if that person will develop during the ICU stay a clinical complication. Yes or no. So we treat it in fact as a classification problem. Will this person develop sepsis over the next X hours? And this we did for a number of clinical complications: circulatory failure, kidney failure, sepsis, to name 3 examples here. We also, and that's a bit of a combination between these deep learning approaches and multivariate machine learning approaches that we used here, and the more classic biomarker approaches, we also did biomarker search for long COVID. And you may have seen this science paper last year about the complement system being dysregulated in long COVID patients. And this is like one of our most impactful biomarker discovery studies that we did. This somehow combines classic and modern approaches of biomarker discovery. But one more example that I want to at least summarize here in 1 minute is that we tried to predict antimicrobial resistance using machine learning from multi-TOF mass spectra, so from protein fingerprints in a pathogen. I'll cut this very short now in the interest of time. So we did this collaboration with microbiologists from Basel, which told us that for every pathogen, a multi-TOF mass spectrum is determined in the hospital to identify the species of this pathogen, and then experimentally the antimicrobial resistances of these pathogens are determined, which takes another 1 to 3 days. And we wondered whether we could, after the mass spectrum is determined, directly predict the resistance profile. And we could with roughly 0.8 or 80% area under the ROC curve, we could predict whether a pathogen is resistant to a particular antimicrobial or not. But, and this is a huge caveat in the modern approaches that use deep learning or at least multivariate machine learning, but there's a huge caveat in this work. In an early version of this work, we could even reach 90% area under the ROC curve. And then we looked at the mass spectra, and we looked at which peaks in the mass spectrum were being used for doing the predictions. And our collaborators told us that there are some uninteresting peaks that were just proteins from certain resistance plates. And we dug a bit deeper, and we noticed that there is like a statistical artifact in the data. Some of the measurements came from a particular unit of the hospital, from hospital hygiene. And in hospital hygiene, they grow all pathogens on resistance plates, so only resistant pathogens survive there. So there's this bias in the class labels in these hospital hygiene data. And our ML algorithm detected proteins that are typical for these resistance plates, and then used the presence of typical proteins from resistance plates to predict antimicrobial resistance. Of course, it has nothing to do with the causal mechanism why there is antimicrobial resistance, but like this, it could boost the area under the ROC curve by another 10%. We only discovered this by human inspection of the features that were being used in these approaches. So this is a caveat of the modern approaches. They are very powerful in predicting, but you have to be very careful that they are not picking up confounders to make these good predictions. Well, we have a huge data set now public, which you can on which you can try out your own algorithm for this task of antimicrobial resistance prediction. It's very important to validate these algorithms across different hospitals and laboratories. We even showed in the paper that it's very important to validate these predictors across time. So as the training data got older, the predictions got worse in our study here. We also showed that if you use multimodal data, I think this is also very relevant to this workshop, you can boost the classification performance further. So we could improve the area under the ROC curve by 12% if we also used chemical information about the antimicrobials. This takes me to the end. So I've shown you in machine learning for biomarker discovery, there are classic approaches that have a high interpretability, but problems with high dimensionality. In the current approaches, we often have high predictive power, but at the same time, low interpretability, and they may pick up hard to detect confounders, and it's very hard to detect this and to find this out that your model is not using them. So what is very important, in my opinion, in present research and in the future, is validation, statistical validation, experimental validation, in medical informatics, also clinical validation. It's very important to collect huge data sets from different sites and to build these foundation models that are very popular now. But even in those, you have to beware of confounders and that they are not just including the right confounder for the right subset of the data. And of course, and this is also at the heart of this meeting today, mechanistic models could be a way out that you really understand which feature, which component of the system influences the phenotype in which way. And here I believe that the tighter interaction between bioinformatics and bioengineering is essential for the future. In fact, in Munich, we just got a very large national cluster for doing exactly this kind of work, like collaborations between bioinformatics, biophysics, and bioengineering. So thank you very much for your attention. I hope that I could share my view of biomarker discovery but I also want to stress that there's enormous potential to solve long-string biomarker discovery problems using machine learning. They are not only problems, they are also huge opportunities. Thanks to you, our funders, and to my team.
That's great. Thank you. Thank you so much, Carsten. This is a lovely presentation. I have lots of questions myself. We don't have time to ask them live, but I am asking everyone to please post your questions on the chat, please. So without further ado, I will just share the screen again just to quickly introduce Fei-Xiong Shen from the Cleveland Clinic. He's a founding director of the Cleveland Clinic Genome Center, a systems biologist, and specializes in genomics, extensive expertise in developing and applying various systems biology and genomic medicine technologies. His lab focuses predominantly on Alzheimer's. And today you will hear about his talk, which is leveraging genetics, multi-omics, and real-world patient data from diverse populations, and how it fuels drug development in Alzheimer's disease using scalable Gen AI models. So I will now pass it on to Fei-Xiong. I think you can share your screen. Brilliant, thank you.
Can you see my screen?
It's loading. We can see a black screen at the moment.
Still loading? Okay. Let me just reshare. Interesting. Why it take us so long? Okay, apologize. Let me share again. You still cannot see my screen, right?
We can see just a black screen.
Okay, but I started the sharing.
Now we can see it.
Okay.
Great, thank you.
I just switched computer, so I have backup plan, so good, right? So yeah, good morning, everyone. Thank you so much, right? So I'm the Vision Chain from Cleveland Clinical. I'm also the co-leader for this, AI, alliance, for AI for drug discovery group as well. Today, I just briefly share our recent work, how we leverage the AI in particular, AI ML approach for both target identification and drug discovery. Use the Alzheimer's as an example. I'll also try to cover other diseases, just show some example how we can utilize AI for drug development as well. So this is my disclosure. So lab mainly focus on Alzheimer's disease, like most of you know. So this is the data we summarize every year with my great colleague, Dr. Jeff Cummings. This plot just to show the landscape for current, Alzheimer's drug discovery pipeline, in 2025. But we learn from this pipeline is Alzheimer's drug discovery is pretty challenging, compared to other complex diseases as well. But the last couple of years, like lecanemab and duocarmazine was approved in the last couple of years, really encouraging news for our field as well. So this is why I call my lab about 7-8 years ago when I built my independent lab in Cleveland Clinical. I call my lab more focused on Alzheimer's medicine discovery. So our goal or mission is try to create both AI and also multi-omics technology, focus on both target and biomarker discovery, and also drug development as well. So the right figure really just show a good example how we build this kind of computational model from the patient data, both the clinical data and the multi-omics data we can leverage, from different cells. And beyond the computational work we did lab also do some wet lab, include both patients, stem cell iPSC derived cell model, animal model, also large scale EHR data validation. So if we get access in both AI and also experimental, in particular for human model, these kind of findings we hypothesize more likely to move to the clinical testing with a potential success as well. So in the past few years, lab was in particularly funded by National Institute on Aging is part of NIH. We are part of Alzheimer's disease sequencer project we call AI ML consortium, and since last year I'm the co-chair for this consortium as well. So our mission for this consortium is really trying to transfer large scale or massive human genetic funding, also multi-omics fundings for drug target identification and drug development. Just focus on Alzheimer's disease as an example. So for example, a couple years ago, did by very smart, faculty, Jillian Xu in the lab, she helped us develop this approach we call NATOTech, is interpretable deep learning and a multi-omics, approach. So use this approach, we can prioritize possible drug target, for, follow up, experimental validation. In this study, she also identified a few, possible drug target we can validate or we validated in our, larger scale EHR data as well. So beyond this kind of deep learning model, lab also work on a lot in network-based approach. We call like the protein-protein interaction network-based approach. We call network medicine approach. This work probably a few years ago. I really interesting study is we trying to prioritize a possible repurposed drug approved for other disease, may potential for Alzheimer's disease treatment. We systematically screening about 1,600 FDA approved drug to see how this drug can specific target both amyloid and tau pathway. Use this in silico network-based approach. And we come out of Seginifo is we also called Viagra, is a potential drug, candidate for Alzheimer's. And when we look at the real world patient EHR data, we find if individual take Seginifo after we adjustify all possible confounding factor like age, sex, and other disease comorbidities, we find that people around with a 30 to 50% reduced risk of Alzheimer's disease. And in follow-up study, we also mechanistically to validate that Seginifo may reduce the tau hypophosphorylation in Alzheimer's patient iPSC-derived neurons. Also in our recent study in transgenic mouse model and organoid model as well. To put all this data together, we are conducting a small pilot trial to further to do the target engagement for sildenafil before we can move to large phase 2 trial to test possible efficacy for sildenafil in early Alzheimer's patient or even mild cognitive health patient as well. So beyond just repurposed drug, we also work on some other type therapeutic hypotheses like a brain gut access. So for example, here we test a very simple idea how a small molecule like a gut microbial metabolites are in our human body like a gut already, how this metabolite may have prevention effect for Alzheimer's or other neurodegenerative approach. So we use a classical like a machine learning approach, just focus on GPCR, G-coupled receptor as an example. What we found here is we found a few, interesting gut metabolites, very small molecule can have good brain penetration properties. They can significantly target some Alzheimer's related GPCR, and we further validate in our iPSC-derived neural models show that this kind of small gut metabolite can significantly reduce the tau phosphorylation as well, really show the proof of concept, the protective effect for this gut, metabolites as well. But for today, I want to, spend the next few minutes really show one interesting work lab just work on right now, how we use this kind of, idea. I think last couple of years, people proposed like a virtual cell, AI foundation model for human complex disease study. In this study, I really thank you, this very smart, young team in lab or in our genome center. Help us the first step with the larger scale human brain, single cell or single nuclear RNA sequence data harmonization, use this generative AI, framework we here. Use this kind of generative AI framework, we can significantly reduce, the bench effect, the data knot, or many other issues, all this data together from the multiple cohort or multiple study as well. Beyond this kind of, important RNA sequence, transcriptome data harmonization, another key study also work on the clinical data, harmonization as well. Sorry. So, so in this, human brain, cell atlas, we totally, assembled more than 2,200 samples, with the deep phenotype data, disease progression data, neuropathology data, also APOE genotyping data, or other deep, clinical metadata as well. We spend a lot of time with our neurologists to focus on, data harmonization, for this data for downstream analysis as well. So here, just to show the global UMAP plot, the left figure just to show the global UMAP plot for 40 million, single nuclear transcriptome. We assembled from 2,200 individuals, and we highlight some major cell type, including neurons, microglia, like glial cell, astrocytes, OPC, oligodendrocytes as well. I would say this is most comprehensive so far, the single cell transcriptomes largest in Alzheimer's domain. And the right figure just to show deep phenotype data, even level 3 data for like in particular for neuron as well. One interesting neuron we highlight here, for example, inhibitor neuron, may associate with a protective effect for Alzheimer's as well. So during the time, I really want to just give, one cell type, we are interested in, or many other groups we are interested in as well, we call microglia. Here, just to show about 700,000 microglia nuclei or single cell transcriptome. And use this, larger scale microglia, transcriptomes, we can look at the different microglial states, use this, trajectory analysis or other, modeling approach. For example, we can see here clearly the homeostasis microglia or even DAM, like disease associated microglia or with the inflammation microglia or other type, microglia states. And our microglia, map resolution is better than some, other small cohort study, overall. But the key question for us is how we use this, complex map for therapeutic discovery or prevention. So one idea we think about here is how we can build this AI model or even network-based model to identify some key gene drivers or like molecular drivers, can involve a different cell type, translation, for example, from homeostasis microglia to inflammation microglia. If we can block this, process, we may can reduce the disease or even, significantly reduce the disease progression as a microglial targeted therapeutic approach. So one interesting I really wanted to share with you, just use the DAM, like a disease associated microglia as an example. When we look at the Alzheimer's patient brain, and also another interesting comparable we call the brain resilience. Brain resilience means this human brain have strong amyloid or tau neuropathology, but their cognitive is still healthy when they die. We call the brain resilience. You can see, use the DAM as an example, they show very different trajectory, for AD patient brain and a resilience brain as well, in both, women and men as well. But one interesting protein we highlight here, for example, USP18 are, key negative regulators in the type 1 interferon gamma signaling pathway, a key innate immunity pathway. We see more significant in, women, but not see in the men as well. In our another, ongoing, collaboration project with Miho from IBM, as well, we took a very simple idea, try to identify repurpose a drug. This drug can block the transition between like a non-inflammation microglia, like homeostasis microglia, to the inflammation microglia. And we predict like catalytic is top drug can block this process. And then we collaborated with Miho to test the large scale market scan data, database. What we found, his group, like, Michael or other found that, if female or woman individuals take the catalytic, they significantly associated with a reduced risk of Alzheimer's. But when we tested the men, we didn't see any difference. And our lab further to test the catalytic in our patient iPSC-derived microglia models, we found the catalytic can specifically block the interferon gamma signaling pathway we show in the previous slide. This is just really one example how we take some cell type or even microglia targeted therapeutic hypothesis, and then we can validate in the real world data, and also human model as well. So next minutes, I want to switch another topic to maybe share with the audience about a more general drug discovery, use some generative molecule design approach in both small molecule and large molecule as well. For example, last year with my great colleague, Dr. Hai Yuanyu from Cornell University, we are developing a deep learning approach, like a pioneer approach. We can systematic way to predict the protein-protein interface interaction, like the first keynote speaker used the same type of approach to predict the protein-peptide interaction as well. The pioneer just to show how we utilize both structure and a sequence deep learning model pioneer to systematic way to predict the protein-protein interaction interface. The middle just to show one example how we can build this kind of 3D interface model for E3 ligase as a key degradation model for cancer as an example as well. And we see many key cancer mutation or driver mutation may significantly enrich in this E3 ligase protein interface as we predicted by our pioneer approach as well. And we further validated using the KEAP1 and NF2 as an example. What we show here that the key protein-protein interface mutation we predicted use our pioneer approach are significant, changed, when we see in the co-IP experiments. And this change or perturbation also associated with the, cancer survivors, clinical survivors, just use the, non-small cell lung cancer as an example. Another, work, beyond just this protein language model lab also try to build a different, foundation models. For example, here we build a, try to consider a small molecule conformation change, use some, computation vision approach. We call molecule, video derived foundation models to help, scientific drug discovery as well. So this approach, we can consider the 3D structure conformation as well, like I mentioned, in the beginning. So beyond this video mode, lab also develop other type approach. Use, the image mode just consider like a 1D and a 2D, dimension to assemble more larger scale, molecules. For example, here we build this kind of, pre-training model, use the 10 million molecules, drug-like molecules, we assembled. So image mode compared to video mode is more computational efficiency to build a more larger scale, model for small molecule, screening the enemy properties prediction or even target, identification we validated in, COVID-19 as an example. So Next, I just want to use a quick example to show that, just use one example, like the second speaker also mentioned, use the, drug resistance or even antimicrobial resistance as an example. We built this kind of, peptide foundation model, use about, 50 million, peptides available in UMAP Cloud, just work on the small peptide to build this kind of, peptide foundation model. And then we try to predict, some, high potent, anti-microbial, peptide as, against the drug resistance as well. Here, just to show overall, foundation model we did here. So in this foundation model, we focus on two key parts. One is focus on the pharmacokinetics, we call the antimicrobial, potency. The second one work on the peak, pharmacokinetics, because most of the drug resistance come out of the bacterial membrane, patrician properties or transporters. We want to design a peptide can pass the bacterial membrane very well, and we also validate in both, in vivo and mouse model as well. Just one example I show here. So used the penetrating is a well-known peptide as an example, and we did two optimization. One optimization or focus on the potency, I mentioned before. The second one work on the pharmacokinetics to improve the design of peptide, to pass the membrane very well. And the right figure just to show our, two-runner design, the peptide, UMAP Cloud, overlap with the existing peptide, available in the market as well. But surprisingly, in our fourth runner design, we did the in vivo, experimental validation. About 88% of peptide we choose for validation have, better, efficacy compared to the existing peptide as well. We did a second run, design based on two peptide. We, have a strong, binding on, potency. We get a 90%, from the 11 peptide we synthesize, and 10, peptide have much better, potency, and also can, against the drug resistance as well. This really encouraging us to apply this kind of, peptide foundation model to identify potent, peptide, anti-microbial peptide to against the drug resistance as well. So beyond just this kind of more AI work, lab also work on some, just recently, work with IBM together, just our IBM Cleveland Quantum One to do some exploratory work, just do very, simple work. This to just on a review. Use this kind of, quantum, computing, framework. We can predict the protein structure in a small scale. And what we show here, just this kind of, RMSD or even binding affinity we predicted in our, Quantum One, also comparable or even better than AlphaFold3, just in the small scale as well, during the computing, powers. And we are very excited in this field. Hopefully we can show more, interesting finding in the next few years as well. So the goal, future work, is really trying to build an end-to-end AI drug, driving a platform for drug discovery. Use Alzheimer's example, anti, resistant as an example. We also explore other complex disease we're facing in the healthcare, system as well. So we're happy to open for all cooperation under the AI, alliance, for AI, for drug discovery group as well. So finally, I really want to thank you, my lab members to, hard work to help me to present here, and also thank you all my wonderful, colleagues here, Miho, Jane, Diana, or many others for our AI for Drug Discovery Alliance as well. We are very happy to continue this collaboration as well. So I just stop here. Thank you.
Thank you very much. This was a great talk, and great first three sessions, three keynotes we've had. Any questions? We are into the break time at the moment, but we can perhaps take one question if anyone has one for any of the three panel speakers. You might have to type the questions on the chat so that we can see it. Not everyone has access to the microphone, but we can grant you access if need be. Okay. And while people are thinking about their questions, we do have a few minutes for a break now. And we have the second session after this one. We'll have another three exciting talks about 3 different topics with speakers from NIH, Sanger, and Merck. And so very much looking forward to those. In the meantime, please do post your questions on the Slack group or on the channel here and channel chat, and we'll be very happy to get your questions answered. So we'll just take a quick break now, and we'll be back very shortly at 10:25 US Eastern. Thank you. text imaging processing, AI, and machine learning. In addition, Dr. Liu is the adjunct professor of computer science in University of Illinois Urbana-Champaign and associate editor of the journal JAMIA and Bioinformatics. Today, Dr. Liu will talk about a recent Nature Methods paper that his team published. His talk is on Gene Agent, Knowledge-Grounded AI Agent for Gene Set Analysis and Biomedical Discovery. So please, Dr. Liu, if you can share your screen.
Thank you so much. Can you see my screen?
We can, yes.
And you can hear me okay?
We can, yes.
All right, wonderful. Thank you so much again for the kind introduction and invitation. So as introduced, I come to you from the National Library of Medicine, which is part of the NIH, and where we are within the intramural research program. I lead a research group interested in using AI and machine learning for both biomedical text and image processing. So on a routine basis, we're primarily working with text data that includes, for example, millions or tens of millions of papers in PubMed and PubMed Central. When access is given, we also work directly on patient data that includes EHR, free text in clinical notes, as well as patient images, in particular in ophthalmology and radiology, where we have wonderful collaborators on the NIH campus. So today's talk will be focusing on some of the recent work related using large language models in biomedicine, and in particular ways that we're thinking about how AI can be used to accelerate drug discovery process. There are two things that we're mainly focused on the gene agent, but if we're interested, I think that ChatGPT, that's our earlier last year's work, is also very relevant for drug discovery. But before I talk about gene agent for gene set analysis, I want to go back to talk to something that I think everybody is familiar with, because even though AI is a very hot topic today, but since day one, I've been at NIH, which is about 19 years, 20 years now, we're always thinking about using AI and machine learning for improving PubMed. Because the unique position that we are at within the NIH. So this is a particular use case that I want to highlight here, is that you can see from this slide a list of different search assistants. These are search tools or search assistants that were exclusively implemented using AI and machine learning. Now, one thing that differs from us compared to commercial search engines or general search engines is that we are very transparent about what we do or what we implement. So in fact, a few years ago, we even write a paper about what's exactly happened under the hood and how our users are responding to these computationally generated search aids. But I will say that today's scientists and like myself, my main challenge in literature is not that I can't find papers. I have some information need. I can usually come up with 3 or 4 keywords. The problem is that when I give these 3 or 4 keywords into PubMed or any other search engine, I'm returned with hundreds, thousands, sometimes tens of thousands papers back. So for someone, an individual researcher like myself or like you probably can't afford the time to go through that long list of papers to synthesize information and to make the connections that are maybe hidden within the large set of papers. So the million-dollar question is that how can we teach computers to understand the papers just like the human scientists, and then they can scan a long list of papers very efficiently. So towards that goal, we have made some baby steps. We have developed tools using natural language processing and literature text mining to mine the literature about highlight the important concepts and their relationships between these key biological concepts like genes and diseases. So we also not only developing the NLP algorithms, but also building web servers that can facilitate access to these semantic annotations. So some of what I'm showing here on the left, there are 3 different web servers. Some are very disease specific, like the COVID system that was built right after the beginning of the pandemic and index only the papers about COVID-19, versus Litvar is really a database or server about genotype, phenotype relationships in the scientific literature. And different from these domain-specific or disease-specific websites, we also have one called PubTator, which I often call it PubMed on steroid, because it indexed all the papers, not only in PubMed, but also in PubMed Central. So it's already a unified search space. But in addition, we have annotations that I show you on the right side, where we highlight the diseases, genes, drugs, chemicals, human species, cell lines, and their relationships. So together, these databases has been actually widely accessed, and one of the important use cases is actually drug discovery. So a lot of the farmers actually download our data or using our APIs to access these semantic annotations. But I guess the main reason that I'm invited to speak here is because of our more recent work with large language models. And everybody to this audience, no need introduction to large language models or ChatGPT, because everybody probably already using it. And not scientists are using it, but general public this is also a household name for the general public. My wife, who's not a scientist, uses it, and my kids are also using it. Whether or not they should, that's a question, that's debatable, but they are using it. So compared to many other very popular online applications, it took only 5 days for ChatGPT to grab 1 million users compared to Netflix and others, for example. So if you are paying attention, you probably will see a lot of news on the AI these days, especially in biomedicine. And there are generally 2 camps of the news on this. One is showing how great they are, what are the potentials and benefits they can bring into healthcare. But at the same time, more recently, especially, there are increasing number of reports showing the limitations as well as risk and danger of using these AI agents or chatbots in healthcare, on patient care. So in my lab, I think both are legit, because in my lab, we actually looking at both areas. So on one hand, which is on the left side, we look at the importance of using AI for important biomedical applications. And on the right side is one of the studies that we published last year, looking at the potential danger, potential limitations and challenges of these most powerful AI models. Because in my mind, today's AI is really getting expert level performance, and because that, it's more than ever important to look at their potential limitations, because they're at the edge of being deployed into real world applications. So today's focus will be on the gene agent, because this is a study that we recently published for gene set analysis, which is important for drug discovery. Because for those who are not familiar with gene set analysis, the input of the system is basically a set of genes, and that can range from tens to hundreds of thousands to tens of thousands. They are generally generated or derived through these high throughput experiments, for example, genes that are differentially expressed under different conditions. And the goal, expected output, is the collective function of this set of genes collectively. And because we're not looking for genes of individual genes in a set, because in that case, you can simply look out for these databases for their functions, but we are rather looking at the functions and pathways for that set of genes playing together. And and these are important because, like I mentioned, they are important in revealing the mechanisms of diseases as well as for drug discovery purposes. And the go-to method for gene set analysis is that using pathway analysis or using enrichment analysis. But they are fairly successful. But the limitation of such a method is that there are two main limitations. One is that if you have your set of genes that are very novel, because existing methods rely on what has been curated in the existing knowledge bases. So if your set of genes is very novel, has very little overlap with existing curated knowledge, then you're sort of out of luck. In addition, the current method do not actually can provide an explanation. They can give you the prediction about the function for the set of genes, but they cannot tell you why these genes and how these genes play together for that particular function. So in late 2023 and early 2024, there has been some pioneering studies showing that you don't have to do much. With ChatGPT, with large language models, you can just give that set of genes to a large standard off-the-shelf large language model, and they can already give you the predictive function, and to the same accuracy as those traditional pathway analysis and enrichment analysis. This is quite amazing. And under 3 different benchmarks, they showed that ChatGPT, in this case GPT-4, was the best, will give 60% of the time to the answers that are similar to the ground truth. And then in the remaining 40%, 1/3 of time, they match a function to the broader term in the ground truth. And then the other 2/3 are errors or sometimes are hallucination. That's the nature of large language models, one of the limits. So we're thinking, can we actually not taking directly the predictions, but do some further verification to improve the reliability of the prediction results. Now, one way to do that is you invite human experts to look at the predictions by the first large language model, and then based on their domain knowledge to revise if necessary, and then make the corrections. But this is very time consuming and perhaps it's not scalable at all. And the other option is you can invite a second large language model to serve as a judge. Obviously, this is a very scalable approach, but researchers have also shown there can bring biases if you're doing that, because one way of bias is that there's a tendency that the second large language model tends to agree with the first large language model. And in that regard, they tend to also focus on text coherence rather than fact checking during their verification. But we still like this approach because this is the only way that we can make this correction, this analysis verification at a scalable, in a scalable way. So what we did, or the idea is, we want to say that if the second large language model, when they doing the verification of the results of the first large language model, can they check against existing knowledge in our domain? And indeed we do, because we're in biomedicine. There are a lot of expert curated knowledge bases about individual genes or about set of genes, even though they may not be represent the entire set of the input gene, but they can be a subset of the input gene. So with that idea, and also the hard work from Dr. Wang, a postdoc fellow in my lab, we came up with this idea called gene agent. Basically, that's highlighted in the red box here, is that we're going to use a second large language model to verify the results of first large language model. But we're going to do that when checking with existing knowledge in different various databases that are humanly expert curated. And then we output the final prediction for the input gene sets. And here's a more concrete example where I show you on the right side, there are 6 genes that are, and these 6 genes, the first large language model, for example, GPT-4 would generate, say, these 6 genes is involved in RTK signaling. Now, given that claim, or given that potential answer, we will send that to the second large language model for verification. And that second large language model will do this in an autonomous way. They would fire different APIs given the set of genes, and then checking out different databases for knowledge that are relevant for these set of genes. And sometimes they may have information for individual genes, sometimes they may have information for a subset of these genes. And then in this particular case, with that information returned from the knowledge base, and then the second large language model will do a reasoning and believe that these 6 genes are not directly involved in RTK signaling. So the original claim is rejected in this case. Instead, it modifies to say that these 6 genes should be more inclined to be involved in the MAPK signaling pathway. So just by doing this self-replication in an autonomous way, we see that on 3 different benchmarks, these 3 gene benchmarks contains gene sets in total over 1,000 gene sets, and on average about 50 genes in each set. And we also made sure that these gene sets are not, the data set, the ground truths are not part of the model training. So that's very important, because otherwise it would not be fair comparison. But just by doing this self-replication step that we just described, as you can see in the results down below, when we compare with the standard GPT-4 off the shelf, we can always gain some improvement using this self-replication. Sometimes it's quite significant within different evaluation matrix when compared to the ground truth. So with that result, we were very encouraged, and through our scientists, colleagues at NLM, we get connected with 2 domain scientists from NCI. At the time, they were actually doing gene set analysis with their novel gene sets that had never been published before from mouse melanoma cell lines. So they give us 7 gene sets from their experiments fresh, and we basically put these gene sets into both GPT-4 as well as to gene agent. And then we, without knowing the ground truth, we returned the results back to them, and they asked the 2 domain experts to evaluate the 2 gene sets, compare them against each other in terms of relevance, in terms of consistency, so on and so forth. As you can see in this slide, where we, for the 7 gene sets, they compared, they looked at the results, they agreed on 5 of them that gene agent performed better. In one case, GPT-4 performed better, and then in one case, they actually have a different opinion on that, so that we can say neither is preferred by since there's no consensus can be reached with each other. Gene agent is nice, but there are certainly limitations on the gene agent work. For example, right now we are only able to perform analysis for human and mouse genes, because that's where most of the database information is focused on. We haven't really performed evaluation on other species. Because of the back-end large language model we use, we know we are able to handle tens or hundreds of genes, but not necessarily thousands of genes. So that's something we've been working on, because as you know, if you're using large language model, there's a context window that we can enter the input. And also, we have some follow-up work on this gene agent, because we believe there's one important thing that we can make it even better, is that every users, when they do a gene set analysis, they have some experiments, they have some study design specific in their mind. And adding these contextual information is able to inform they getting a better predictions. Now, just like any other projects we do, we're very much open. We have the website that we would like you to try and give us feedback. If you want to collaborate, or if you want to replicate our study, you can download the source code as well as the data that we use for evaluation. So I think my time's up. I see the moderator here, and I'll stop here, and thank you for your attention. I'm happy to take questions if there's any right now, but this fits the whole umbrella of the kind of work that we do in my lab. We're not interested as academic, we are unable to develop the greatest, the most powerful large language models, but we really want to focus on the ones that are important for biomedicine, and whenever possible, just like the work with the gene agent, we want to piggyback the existing most advanced models, and then making that better for our domain. And like I said, that's on the one end, and on the other hand, we are looking at potential problems and challenges for these large language models in practical applications. Thank you so much again, and I look forward to any questions, or people can feel free to email me here.
Thank you very much, Dr. Liu. This was a very, very insightful talk. I do have lots of questions as well because we have done some work very similar to what you presented, so that's very interesting to us. And if you could please also share with in the channel in the chat any links to the systems that you're looking for feedback from. I'm sure the audience here would be delighted to try them out and send you feedback. So thank you again for your talk. Any questions? Any questions from anyone? Please post them on the chat, please, and we will swiftly move on to the next speaker. So I'll just quickly introduce Professor Hanifa is a dermatologist pioneering the applications of single-cell genomic technologies to understand tissue homeostasis, immunity, and disease pathogenesis. A major research goal of her lab is to decode the developmental or functional maturation of the human immune system. She's the Wellcome Trust Senior Research Fellow in Clinical Science, the Lister Institute Prize Fellow, and Professor of Dermatology and Immunology. In today's talk, Professor Hanifa will talk to us about big data plus AI leading to biological mechanisms at scale. So please welcome Professor Hanifa. Hope you can share your screen.
Yep, I'll try sharing my screen. Yep. Can Great. You see it now?
Okay, we can see it. Yeah, we can see it.
Yeah. Thank you very much for that very kind introduction and invitation. So I'm going to talk a little bit about where we are in this endeavor. I head the cellular genomics program at Sanger, and the focus of it for the next 10 years is big data and AI towards biological mechanisms at scale. So just to kind of put into context how we've got here, and particularly from the perspective of Sanger Institute, there have been several disruptive technologies. Sequencing was there, led to the Human Genome Project, gave us the Book of Life, the relevance of it, which we all know, how the variants predispose to disease. And then came along the ability to sequence at single-cell resolution, and that led to the global initiative, the Human Cell Atlas, which is an ongoing project, which is to give us the Book of Cells. And the aim was to create a comprehensive reference map of all human cells and to be inclusive and diverse representation of this map and for global benefit for everyone. This involves many researchers. As of this year, there are 4,000 members, more than 100 countries, and this year we've also formed a partnership with UNESCO to enable the endeavor to be representative and also to be inclusive. We've got to the first draft atlas, which was published last year from the different bionetworks that contribute towards the entire human body, and there are 18 of those. You can see all of them here. My primary contribution has been the skin and human development. So using single-cell genomics and also some level of spatial genomics, as it's more recent, we've decoded the prenatal human immune system and published this work. And single-cell biology is very intimately linked with computer science. So a lot of machine learning applications have been weaved in terms of the data analysis of the work that has come about. And really where we are now is at the stage where we've got a good catalog of cells, but what we need is to understand how these cells now form tissue ecosystems. So if we look at cells as Lego blocks, we can look at tissue ecosystems as the dwellings that you can make up from these individual blocks, each providing very different functions. And as we all know from looking at genetics, we've got a reasonably good idea of how genetics predisposes to clinical phenotype and the course, including at population level. But there is a massive gap, which is the context, the cellular molecular mechanism of how genetics predisposes to clinical phenotype. A great example will be BRCA, linked to breast cancer. And we know that, but as more and more individuals have been sequenced, and particularly within the UK Biobank context, BRCA inheritance or BRCA occurrence doesn't necessarily mean that one will get breast cancer. So these cellular molecular mechanisms, particularly within the tissue context and understanding the mechanism is really important, and that is the gap of this end-to-end understanding across skills. So if we look at all of the disruptive technologies I've briefly outlined, and what I think is now really making massive impact is machine learning and AI. And it doesn't just impact analysis, it impacts data generation, and also, as you've heard, how AI can also be used as an agent and co-scientist. So the program is really trying to fill in this gap and bringing in vitro organoid perturbation models, particularly genetic perturbation, so we can go from inference to causality, and to undertake this at scale with AI. So we have three interlinked approaches: descriptive observational data generated from patient samples. These are tissues from human development and disease tissues, using patient cohorts and health records, using single-cell and spatial multi-omics, and supporting that with AI tools, particularly foundation models, which will guide us, because this can narrow the experimental space in which we have to explore, and to then guide which are the experiments that we should be doing in an experimental setting, providing that mechanistic insight, and iterating between these three modules will then enable us to drive forwards new therapeutic drug discovery, and also better insight into the pathogenesis and biomolecular understanding. An approach that we envisage is across tissue and across disease, so this is very much disease agnostic, using the three interlinked modules across 5 areas of brain, skin, female reproductive area, kidney, and liver, and looking really at how developmental programs are conserved across tissues and re-emerge during disease, and also particularly the role of the immune system. And we've chosen these 5 areas because of the areas of faculty expertise. So just very specifically on the perspective of generative AI and foundation models, spatial omics technologies have really made a massive difference. This is an image of the tissue section of human skin, and on top here's the top surface. This is a cross section, and what's been colored here are the niches or ecosystems that are important in atopic dermatitis. This is one of the ecosystems. So here what you can see are this perivascular information, and what you can now do is to see that it's made up of multiple cell types. And I guess this just to illustrate how what a clinical pathologist would see, these shades of pink, can now be added with all of the different cell types and gene expressions. And this is where using generative AI models is really incredible in being able to link the multimodal data, particularly with histopathology, which is used in clinical practice, and also to be able to undertake in silico cell and gene perturbations, both in situ in space and also time, observing what happens longitudinally, say, for example, after treatment. And we've developed a large curated data set, both from in-house data generation and available data set for fine-tuning and applying it in the capabilities with as a zero-shot model. So using it on diseases, for example, that it has never been trained on, or tissues that it's never been trained on. And this is very much work that is led by faculty in CellGen, Mo Lotfalahi, and working together with many of the us in faculty members with domain expertise and bringing different skill sets. We're really applying it in a clinical setting, and I'll show you one example, which is a publication that is on bioRxiv right now, looking at how we can map and reprogram tissue microenvironments with a generative AI model, which we call MinFlow. And this is where it learns from spatial data and essentially tries to work out what are the intrinsic and microenvironment-induced embeddings and define the gene programs that are associated with those. And when it now looks at the tissue section, it's able to identify areas in the tissue which are hotspots, where there are signaling from microenvironment-induced cell states. You can then disentangle that and look at what are the cells that are involved and what are the gene programs that are implicated there. And now you can perturb those cell types and see what happens to the resultant gene expression profile. So this is the workflow for the algorithm, and I'll show you an illustration just based on atopic dermatitis. We show it in a renal cancer setting as well. So here is atopic dermatitis skin, where you can see that that's the epidermis, the top bit of the skin, and this is the epidermis. And you can see these areas that are enriched for microenvironment score, and you will realize that it's associated with the histopathological feature, what the pathologists will say are perivascular inflammation. So you can now look specifically at the different microenvironments that you can define using the generative AI model. And dissecting that further, we can see that this is related to dendritic cells interacting with regulatory T cells and conventional T cells, and also the fibroblasts that are found around these structures, the perivascular structures, involving the various chemokines that the dendritic cells are using to recruit the T cells in situ, and are then modulating the fibroblasts to also support the recruitment of T cells. Now, what's really exciting is you can now delete in silico. So, for example, removing either the regulatory T cells that is in that microenvironment, or to replace the conventional T cells with regulatory T cells. So, overexpression style experiment, but with more cells. And you can see how when you remove the regulatory T cells, the change in the gene expression of the migratory DCs, whereby it becomes much more activated, expressing more chemokines and also Th2 immunity-associated markers. And the converse, if you were to replace the conventional T cells with regulatory T cells, you have the converse occurring, a dampening of the maturation markers, a dampening of the chemokines, and also type 2 immunity associated. So this gives us an insight and an ability to assess what are the different cell types in the tissue that are likely to contribute to the pathogenic process, and we can then test in sequence, deleting the different cell types and looking to see if this is likely to improve the gene expression in inducing it to more of like a healthy state, or actually make the disease worse, narrowing that experimental space so that we can now go in or look at patient data after treatment to see where it correlates with, or go in with an organoid model and start to look at the actual mechanisms in play. So I'm going to stop there, and just to thank my lab, my current lab, for all of the work that's been done. The person who's done the analysis in this is Lloyd Steele, who's shown here, and also acknowledging all of our collaborators and funders. I'll stop sharing the screen and very happy to take any questions.
Thank you very much, Professor Nifa, for the excellent talk. We do have a little bit of time before the next talk, so I do welcome any questions. If people are able to unmute their microphone, they can ask the question themselves. If not, please type the question on the chat, and I will be able to read it out loud or help you unmute yourself. May take a few minutes for some questions to come through. Usually they come through during the presentations. So we have a question already. So from Viba Anand. So fascinating work, Professor Nifa. Can you talk about linking histopathology to single cell data? What was needed? Thank you.
So there are two ways of going about this. Thanks, Viba, for the question. The first is many of the platforms now, or at least the platforms we use, enable you to measure the gene expression on the same section where the H&E has been done. So we've used the 10x Xenium platform. So you can then overlay the gene expression directly, and then you can actually integrate with any suspension data. We use the 5K platform. Again, it's not whole transcriptome, but by using ScanView, we can actually integrate it with the single-cell RNA sequencing data, and then have a better idea of the gene expression profile. That's one way of doing it. Others have used the way of like learning from the molecular expression and the H&E, so much more of like a computational or AI-related approach to link histopathology to single-cell data.
That's great. Thank you very much. We have another question from Jeremy Huang. So thank you, Professor Nifra, for the wonderful talk. Have you been able to validate the results of the predicted perturbation described in your in vitro assays or in vitro?
What we've done in this context for the data that I've shown is to look at patients who've been treated with dupilumab before and after, and look to see what happens to the regulatory T cells. If it is in accordance with what we predicted the changes would be following in silico deletion or in silico addition of those cells. So that's how we validated that with human in vivo data, essentially in vivo perturbation, rather than necessarily in vitro assays, as that's a much more accurate readout.
That's great, thank you.
Okay, brilliant, thanks.
I think that's all the questions we have so far. We may have a few additional questions in the chat, so I will make sure those reach you when we get them. Thank you. Okay, so I think we're okay to maybe move on to the next speaker and the last of the keynotes today. This is Dr. Linsky. He's the Senior Director in Disruptive Technology, Discovery Biologics at Merck, holding a PhD in biochemistry and with extensive experience in computational methods for protein design. We welcome him today as he's going to talk about AI as a partner in therapeutic protein discovery. So I already see that you've grabbed the screen, so yeah, please go ahead.
All right. Thank you very much for the introduction, and thank you for inviting me to be able to speak. Can I check that you can hear me?
We can, yeah.
Wonderful. Okay. So today I'm going to talk about AI as a partner in the therapeutic protein discovery process. My background and my expertise is in structure-based protein design, so that is going to be the main focus of this. Aura gave a really nice introduction to this idea and how this capability has evolved over the years as well. But just to recap and give some context, the purpose of a structure-based protein design is to generate sequences that create or make some sort of biological interaction, or some sort of protein-protein interaction, some sort of protein-small molecule interaction, just some sort of interaction. There are two, in nature, the way that this works is you have an amino acid sequence, it folds into a three-dimensional structure. That three-dimensional structure then makes an interaction with something else, and then you get an intended function. So, the advances in protein design that have come recently have been sort of a simultaneous evolution of our ability to predict structure from sequence, and then next to be able to predict function from sequence and from structure, and at the same time, our abilities to generate sequences to generate structures that have a desired function have also really come together. And if you look at this as a whole picture, in therapeutics, antibody discovery technology has been just so incredibly successful over the past many years. And really, this is what it's doing. It's taking a function and it's going through the generation process and getting you back to a sequence. And we are now getting to the point with these really fantastic emerging generative AI tools that we can start to do the same thing using we can just start to do the same thing computationally. The thing that I will point out, and the focus of my next slide is going to be that not every interaction that you generate means you're developing or that you're creating a therapeutic drug. The context for doing that is a lot larger. So So this is how I will map that out. And so the first slide was the protein design context, which is a part of a larger context of therapeutic protein design. So in a therapeutic context, you're looking for sequences that address some sort of clinical need. Some disease, you have some external landscape that's out there, you have some pathway that you're interested in. And then from there, you have this increasing context, increasing complexity, increasing cost and time, and more and more sparse data as you go. And that, in my view, that context grows from having a single protein, and that's basically a sequence that folds up into a certain structure. And you're interested in the biophysical properties of that protein basically by itself. As you expand the context, you get into the protein interactions and how that protein is having pairwise interactions or binding with other proteins. From there, you get more and more and more complex. So like I said, you might have a protein that binds to something, but it has no function within the cell. You might have a protein that binds to something and has a function in the cell, but it's undesirable because of in vivo toxicity, or it doesn't work in vivo, or it has bad PK/PD properties, and you might have something that meets all those criteria, but is not necessarily a good therapeutic. What I want to highlight here is that problem selection is really important to have downstream success, and secondly, that there is no one person or no one technology that is going to understand, at least in the foreseeable future, this entire context. So the way that the problem is selected, the way that the humans interact with this whole system is vitally important, and that is what I hope to highlight here in this talk. So I'm going to talk over two different case studies that I have been involved in. Both of these involve improving AI ML methods in some way or another. So either improving the methods themselves or improving the impact that those methods have. The first one is going to be on a massive folding and stability data set generation that resulted in improved protein design methods. The second will be on a de novo design therapeutic that took a relatively limited AI ML. I wouldn't even call it it wasn't even called AI ML at the time, but it took a relatively limited computational technology and was able to achieve success on a larger therapeutic context. So in both of these cases, and this is what I'm going to reiterate, if there's anything to take home from this, it's that partnerships are critical when you start to expand the context. So partnerships were critical in improving the AI ML methods in the first part. Partnerships were critical in expanding the scope of where those methods are useful in the second part that I'll talk over. I'm not focused on modern AI technologies. There's a lot of other talks today that are. What I want to do here is more take a bit of a historical peek at how methods have advanced, how some limited technologies have been made impactful through effective partnering. Okay. So the first part is and the challenge in as of maybe 5 or 10 years ago is to create a protein sequence that folds into a highly stable intended structure. This, and these are the days where we weren't trying to design a protein that does anything. We were just trying to design a brick, a protein that was highly stable, does nothing, and by doing that, evolve the methods that are out there so that they can do greater things. So the things that were in the way of this, one, sparse data sets. Ability to quantify to what extent a sequence is folded and how well it's folded, how stable it is, was quite sparse. There was not an easy protein design method that did this. The success rates were pretty low, and they were pretty limited in terms of the types of folds that they could handle. And I also think one of the really big limitations that we have is that data sets only contain proteins that work. Or a lot of the available data sets only contain proteins that work. So you're a little bit biased in what you are able to look at. So this is an example of multiple technologies coming together to advance a method. So the way that this study worked and the way that this experiment worked, and there's a few different papers that use a similar method, but in general, you're using a protein design technology to generate a bunch of sequences. You synthesize DNA for those sequences, and there could be thousands of sequences, and that level of bandwidth, that level of high end, lets you also include negative controls in that sample, which can be quite valuable for the conclusions and the application of the results. So this DNA library in one sample is transformed into yeast cells. The DNA then gets turned into a test protein that is displayed on the surface of the yeast cell, and then that test protein and those yeast cells are subjected to digestion by proteases. The thinking behind that is that stability of the test protein, so an unstable test protein is basically going to be more likely to be cleaved by a protease than a really well-folded protein. So the really well-folded protein, the conformation is going to be held so nicely intact that the proteases will not be able to hold the pieces of protein that it needs to hold in a certain conformation in order to cleave. So this assay measures proteolysis by detection of a C-terminal MIC-tag. So basically, if a protease cleaves the test protein, you end up with this C-terminal MIC-tag floating away, and you don't see signal. If the protease does not cleave, you see signal coming off the C-MIC tag, and this is an example of that at the bottom, or before protease treatment, you see higher levels of intensity. After protease treatment, you see that intensity going down. And this is a mixed sample with thousands of sequences. So when you look at this distribution, you hear at the green part under 10 to the fourth, you're seeing some proteins that are surviving the protease digest, and so those are going to be the better folded proteins. End result of this is we were able to create data sets that quantify the stability of thousands of different sequences. So a really big expansion of the data set and a nice quantification of the data set as well. We were also able to use multiple folds as part of these studies. So together, those things informed better methods. So starting from this very low success rate, we were able to develop better protein design methods for the sequence to structure part of protein design that I talked about on the first slide. With success rates that were sometimes approaching or in excess of 50% folded protein. So here we were able to take. And here I should also point out the negative controls here were scrambled sequences with the same amino acid composition as the sequence that was designed to be folded. So by comparing the negative controls that we expect to be unfolded with the positive controls that we expect to be folded, and looking at the extent of protease digestion of both of those, we can get a quantifiable readout. So that's how we were able to achieve that. What that means, is that these studies took three very different technologies, that require and have three different areas of expertise, and partnered them together to achieve a greater purpose. So in protein design, we started off being able to design sequences that fold, especially good at small proteins, but it was done at a low rate and for a limited number of folds. Because of the way these technologies are partnering, because of these studies, the ability to predict and generate stability improved. Advancing quite a bit. We started with a proteolysis assay that could measure stability, and this had been established in literature, but it was limited in the number of samples that it could handle. It was difficult to run. You have to run all the samples at the same with the same timing. And because this was a pooled format, this was a library format supporting thousands thousands of samples, it really reduced a lot of those challenges. And because we were able to express negative controls on the yeast display system, it also really helped with amino acid composition biases of those proteolysis assays. And I think the really big one has been the advent of oligo pool synthesized DNA libraries. So you can where you can have upwards of 100,000 completely designed sequences in one sample, which allows a lot of different conditions, a lot of different features to be measured, like the negative controls. But they also have their own limitations, where you're limited to a relatively small protein size. It just happens these protein design methods were really good at a small protein size, so these things were very well paired with each other, and really we were able to get the most out of these different technologies. So I did my postdoc in David Baker's lab at the University of Washington, and one of the things that really sticks in my mind that he said at one point was that science isn't done in a vacuum, or maybe it was good science wasn't done in a vacuum. And so I'll reiterate that point here, that advancement in protein design tools in this case was not done in a vacuum. The second part is going to focus on a therapeutic project, and here, this was at the start of COVID is when we started this and conceived this. This is just when everything was about to be shut down. People were nervous, and this was really a very huge challenge at the time. So our challenge here is, let's stop the virus. Let's use whatever capabilities we have, whatever challenges, and overcome whatever challenges we have, and just try to create something that might be able to neutralize the virus. There's a lot of roadblocks in place. Our methods were pretty limited. We felt we needed a non-injected or inhaled administration, and there was always that possibility for viral escape, which would be so it would be great for a publication if we showed a SARS-CoV-2 neutralizer, but it would be a bad drug product, in case the virus escapes or mutates and is still able to enter cells and bind, but it's not. But then the drug stops working. It stops doing anything over time. So bad drug, good binder in that case. So this work was really enabled by some of the structural biology that came out virtually almost immediately after COVID. It was really amazing how quickly the structure work in the literature came out. And the way that, quick refresher on the SARS-CoV-2 is that the virus enters cells by recognizing a protein on the cell surface called ACE2. So it binds to ACE2, enters the cells. The part of so the spike protein on the virus is the part that interacts with ACE2. The spike protein has a domain that specifically interacts with ACE2, and that domain contains 3 different structural elements that are making direct interactions. The ACE2 has 3 direct structural interactions with the viral protein. So this interaction was pretty well understood after seeing that structure paper, and this is something we felt at the time we could address. This is when I was with a previous company called Neoleukin Therapeutics. So the end result of this was that a SARS-CoV neutralizer, which would not be possible without some of the computational technologies, we were able to create that thanks to human computational and lab-based collaborations, which I hope will become clear after this part. So again, this is COVID-era protein design, not nearly at the level of sophistication that some of the tools today are at, but it was pretty good at its own narrow context, and what it was good at was creating sequences that fold into a predefined structure. So the stuff that I talked about at the beginning is directly feeding into this. So the thing to point out is that the human is here, I think, is the human aspect. let me tell you a little bit about what we set out to do. So we knew here that success would mean binding to and neutralizing the virus. We knew that it would mean resilience to escape mutations, so ability to work even after the virus is mutated. And we knew that that we needed something that would protect animals via a non-injected administration. So we set out to do this by creating a decoy to the cellular protein that the virus recognizes, in hopes that this decoy would bind to the virus and prevent it from being able to bind to cells, and then it would be cleared. So we took the structural elements that had that bound to the viral protein. So taking that deeper context, and used human more or less human-guided intuition to place supporting structural elements around them, since these three elements were not enough to create a folded protein. And from there, we were able to build connected backbones that were based on loops found in natural proteins. And then we were able to turn the technology loose on what it was really good at. We were able to use it to design sequences that can fold into that particular backbone. So the picture on the bottom that I have here is a diagram of the residues from those three secondary structure elements above on ACE2 and how that looks. And then so this is a three-dimensional structure of one of our design decoys that recapitulates those different binding surfaces. So really trying to match. And the idea is that if we can perfectly match that binding surface, a viral mutation will prevent binding to ACE2 just as well as it prevents binding or stops binding to the decoy, and vice versa. We also had a really nice advantage here in that the interaction with ACE2 and the virus was relatively strong, so it was already in the low nanomolar range. So we had faith that if we recapitulated this exact binding surface, that we might be able to achieve similar affinity, which would be therapeutically relevant. So we were able to design this protein. We started off with something that was a relatively weak binder straight out of the computer. It was about one out of about 200 designs that we tested had any binding activity at all at the time. We were able to quickly turn that around using lab-based methods that were complementary. So we used a directed evolution method that identified weaknesses in the structure, able to really reduce the KD of the molecule, and then we did a serial duplication, which let us interact with the virus at more than 1 point, and that reduced the KD even more, almost to the limits of detection of the BLI instrument we were using. So we did a neutralization assay after that, and found for the best molecules sub-nanomolar neutralization of the binding to the human ACE2. So we were really happy, and we were able to do that in 69 days. We were able to use yeast display technology as well, combined with some of the DNA oligo pool synthesis methods to study the resilience to viral escape mutations. And here we tested about 1,700 mutations, seeing a strong correlation between binding of the viral protein to ACE2 with binding of the viral protein to the decoy. So yeah, and then finally, we were able to show that this molecule protects against lethal SARS-CoV-2 challenge in hamsters and in different mouse models. So that is highlighted. I mean, maybe the one to look at is down on the bottom, where as the study progresses, animals that received the decoy showed a higher chance of survival, and it was a dose-dependent response. So really excited about that. And the thing I'll come back to is we had very limited computational technology here. And one thing that I asked myself quite a bit is, what would this look like today? And so this was a 2020 study. We were able to do all these things. Just for fun, I ran this through a more modern workflow using RF diffusion, protein MPNN, and tried to see what would happen. And you come up with almost the exact same type of structure from this. If I go back and think about how this was done, I think the result using the modern tools would have been quite similar. You would have had sequences that are more likely to fold and function, probably. You would have had very probable earlier identification of the different problems with the proteins that increased this time to get to the composition of matter. But ultimately, the final molecule probably would have been quite similar. And what that highlights is that problem selection is quite important. We chose a problem that was very well suited for the limited computational technology that we had. And that's something that in today's world is still going to be a thing, because nothing understands the full context behind drug design. The other thing to point out again is that effective partnerships were truly enabling. Maybe the most important thing of all those being a thorough understanding of the strengths and limitations of all partners. So including humans, including computational technology, including the lab-based technologies, and so being able to select the right path to solve the right problem at the right time. So I said, yeah, the results will be kind of similar, but the last question I'll leave you with is, for how many needs would this have worked? So this is a very, very narrow problem definition. And with today's emerging technologies, I think we can solve a lot more. And that's really where some of the benefits of that come in. So yeah, take homes. You don't make advancements in a vacuum. You need close partnerships across many different domains, especially for something as complicated as a therapeutic concept. AI/ML technologies can expand a set of addressable problems. So increasing the scope of problem by which, so the study that I showed earlier has a narrow scope, very narrow problem definition. We can address a lot more problems now. This technology augments people. It augments experiments. It can accelerate speed, increase chances of success, but it also doesn't solve everything. It doesn't replace experiments, especially in those later areas of drug discovery where data is particularly sparse, and it doesn't remove the need for people and guidance. And today, all those things apply. So these are all the people that were, speaking of collaboration, these are all the people that were directly involved in the couple of papers that I shared with you today. I'll thank again today's attendees and the AI Alliance for the invitation to speak. Thank all the people that contributed directly to this, and especially thank all the prior work that this was built on. All right, thank you.
Great, thank you very much for your presentation. This was very, very interesting. I again have lots of questions, but I'm going to open up to everyone in the chat, and those who can unmute themselves to please come forward with a few questions. If not, I have one. I know we're running into the break time a little bit, but if anyone's happy to stay and engage with some questions, we can do that. So I'll just wait a minute for some questions. Yeah, hi.
I had a question. My question was, I guess, first of all, in the way that you did this, the last project, how did you actually, you said you connected the backbones based on loops found in natural proteins, and I was just wondering how you did that. And then also, I think you said you didn't want it to be an injectable, so then was it intranasal? Is that how you avoided the degradation problem?
Yes, yes. So let's start with the second question first. Yes, it was an intranasal delivery. We were aiming for a spray-dried formulation and an inhaled powder at the end of it. What we dosed the animals with was an intranasal liquid. And the formulation and the intranasal, which I don't think I mentioned, but that was the reason why we went with a de novo protein modality, is so that we could get that extra stability that you and solubility that you would need with those approaches. Wild type ACE2 did not, it doesn't have great properties that would be amenable to those things. And then your first question was about looping the proteins, which is a great question as well. We had developed a proprietary clustered database in-house. So we had, we basically took all the known loops within the PDB, clustered them together, and there are a lot of common themes that emerge when you look at how nature connects secondary structure elements. You can then take the ends of each cluster center, look at the 6D transformation in Cartesian space, and find ones that match most closely with the disconnected secondary structure elements in the design backbone. Does that answer your question?
Yes, great. And great job.
Thank you.
Okay, and I also have a question a little bit more generic and a little bit tongue-in-cheek as well, but just, well, first of all, you were talking about partners as they're needed, right? So several partners needed to help look and understand this full picture. You did mention that no one person or AI can completely understand the entire therapeutic context, and I do agree with that. But I would challenge that slightly. I think some people in the audience, particularly those with a technology background, they're currently building AI agents, they may be thinking, could we build AI agents and agentic systems with a human expert in the loop, or maybe several human experts in the loop, to help address this and to help broaden our understanding of therapeutic context?
Absolutely, I think that is true. And that was part of my aim in calling out partnerships in this way. It's with an eye towards those agentic AI methods. And the more cases we can get where human involvement and playing to the strengths of technology are out there, the better we can make those methods. And I qualified that statement in the, at least I hope I did in the talk, by saying yet, or not right now. And I think in the future that will certainly be that will certainly be a thing, and I'm very, very excited to see what the people in this group are able to do in the future.
That's great. That's a great answer, and really motivating for the next sessions that we have later today. So we do have a few minutes break now that we can take. So about 3 minutes. We're supposed to start again. Thank you so much again, Tom, for your talk. So we'll take a quick break now, and the next sessions, one of them there will be separate sessions in 2 breakout rooms. One of them will actually talk about AI agents at the frontier of biomedical research. The other will be a series of walkthroughs of open source tools and models for biomedical research. So we'll start at 11:35 US Eastern. That's 16:35 in Western Europe, and the other times as well are on the slide there. So we'll take a quick break now. We'll be back in a few minutes. Thank you. Okay, that was a very short break for some of us, but I hope everyone's feeling okay to proceed with the current time that we have allocated for session 3. And I will be setting up 2 rooms now, so 2 breakout rooms. You will have to choose the room that you want to join yourself. So we'll give maybe a minute or so for people to be joining the rooms. So room 1 will be a walkthrough of open source tools and models for biomedical research, and room 2 will be about AI agents at the frontier of biomedical research. So the rooms are now open. I will be chairing room 2, so I will move to that room. I think Efrat Exter will be chairing room 1, so Efrat, you could maybe move to that room as well. And then each of the moderators in that session would be recording those sessions independently. This part will not be broadcasted live on YouTube and LinkedIn, but there will be a recording of the 2 sessions. They'll be done separately. So I will now move to room 2, and we'll get started. And then we will be back in about an hour's time with the panel that we have in about an hour's time exactly. So thank you.
Association. improve the prediction of drug absorption, metabolism, exertion, and toxicity. And before his current role, Pat spent 30 years in leadership roles at Relay Therapeutics and Vertex Pharmaceutical. So thank you very much, Pat, for joining us. We have Tomer Cohen, who earned his BSc and MSc in computational biology at the Hebrew University of Jerusalem, and he is currently pursuing PhD under the supervision of Professor Dina Schneidman, who was supposed to join us but couldn't. So thank you, Tomer, for coming in. His research focuses on modeling antibody-antigen complexes and on de novo antibody design by leveraging recent advances in deep learning. And we have Aled Morgan Edwards, who is the founder and chief executive of the Structural Genomics Consortium, which is an open data generation organization in the area of protein and chemical biology. And he is also a professor of medical genetics and medical biophysics at the University of Toronto. Thank you all for joining our panel. And I'll start by asking you, maybe we'll start by the order I just shared your name, so maybe we'll start with Pat, if that's okay. Can you tell us about your journey toward benchmarking in biomedicine? What sparked your interest, and how did you become engaged in this space?
Sure. I've been developing methodologies for a long time, both physics-based methods for making predictions in drug discovery, as well as machine learning models. And really, the only way I know to validate these sorts of models is through prospective predictions. So initially, I think by accident, I ended up getting involved in an initiative that the American Chemical Society had called TDT. And then after that, I ended up getting involved with some people at UCSD who were working on D3R. And then, you know, it's once you start working on these things, people are like, oh, you worked on that one. Why don't you come work on this one? So I worked on CASP. I worked on CASP-15, the ligand prediction challenge. And then more recently, I've become a part of OpenADMET, where we're putting together data sets which we can use to sponsor blind challenges and allow people to make predictions with a variety of different methods.
Thank you. That is very interesting. I have a lot of questions on all what you already have said, but we'll go by order and just hear first from everybody. So I'll move to Tomer. Tomer, can you say how you got involved in that?
Yeah. So for me, it started when we developed tools for antibody design and folding and docking. And when you wanted to publish our paper, we need to do benchmarking of all of our tools. And I think the first thing that we discovered is you compare benchmarking in antibody folding or design to other machine learning fields like images, we're really missing these benchmarking data sets that are the same across all papers. So my interest started from this challenge of building our test set, and then also seeing that every paper uses different benchmarking tests, so metrics. So this is how my interest started in this, and understanding that there is really a really great need for this general benchmark for antibodies, and I think biomedicine in general.
Thank you very much. It sounds like that you are following this approach of jealousy among scholars increases wisdom. So you saw the CASP, you saw that they have a very robust way to benchmark, and you said, hey, I want also to have a robust way to do it. So that's nice. good to know that there are different circles and different way by which benchmarking in one area influences a nearby area. Thanks for sharing, Tomer. Aled, are you willing to share with us?
Yeah, sure. So my interest in this emerged in the mid-1990s when we started to solve crystal structures at scale in these so-called structural genomics efforts. And our teams at one 5-year stretch were contributing about 20% of all the world's novel structures to the protein data bank. We had a real focus on data quality and on contributing proteins to CASP. And I think John Molt says our teams were the largest contributors of structures to the CASP competition. So I sort of watched the protein structure prediction thing happen over the last 30 years, mostly as an experimentalist donating our data to the effort. In the early days, also, our team, Cheryl Arrowsmith specifically, solved a lot of structures for David Baker. This is 15, 20 years ago, when they were predicting molecules and they needed to know what they actually look like in the real world. And so, that's how we saw benchmarking work. Now, more recently, of course, there's been an explosion in the interest, but the science, I think, is holding back a little. I don't think there's enough emphasis on data quality, and so specifically in drug discovery, our internal data had shown that the hype was exceeding the reality. So we and 4 pharma partners started a prospective benchmarking and critical assessment thing called CASH, which challenges the community to make prospective predictions about which compounds bind which proteins. And the results have been predictively revealing and predictively underwhelming. But they will get better. And so the only way they're going to get better is if you systematically compare over time. And I think that's what we're talking about today. Thank you.
Thank you. And I'm wondering, Pat, if you have anything to comment about the not enough emphasis on data quality and the prospective testing. Do you have anything from current days that you are also looking at that is aligned or disagree or agree on that?
No, I agree completely. I mean, most of my work has been in small molecules and in predicting properties of small molecules. And if you look in the literature, the vast majority of the benchmark data sets that people are using are terrible. These data sets were curated out of dozens of different papers where people didn't do the experiments the same way. So trying to take this and then build a machine learning model to make predictions and benchmark on this is kind of silly. We need to have large data sets that were consistently generated that cover a realistic dynamic range. And we need to make these data sets available for people to benchmark their methods on. And I think without these data sets, the methods won't improve. Or if they do improve, we won't know it. So I think, this is essential to the progress of the field.
I completely agree. And I'm wondering the fact that we have here all that represents a large not-for-profit, right, the SGC, and Tomer, who is working as a PhD student doing his work, and you, Pat, working in a company, right, that has a business model. It's not a company, it's
No, we're a nonprofit.
Nonprofit as well, okay. So maybe I'll put it as in that way. What governance models or community structures do you find most effective for maintaining and evolving biomedical benchmarks like the ones you just discussed? And do you think it's essential to have such not-for-profits? Or how do you involve? I know that Ali is extremely successful in involving for-profit. You just mentioned 4 pharma companies. I don't know much about it on companies you are involved in your governance process. So I'm very curious to understand how to drive the community. What's your thoughts about it, Pat?
I think the best way to do this is to have publicly supported efforts that will generate data and make that data available. I spent 30 years working in the pharmaceutical industry. I hear a lot of people talking about, why doesn't pharma just open up their data? And I don't think that's ever going to happen. There's too many legal and intellectual property restrictions there. So I think the best way to do this is for us to work together as a community, agree on what constitutes a good data set, have publicly funded methods of generating that data, sponsor blind challenges like CASP and CASH and what we're doing with OpenAdMet, and then use those to bring people together, compare notes, and work together to improve our abilities to do predictions. I think this is something that we all have to do together.
I think the highest appreciation we saw is the Nobel Prize laureate that people involved in these competitions received. So we do see the community is appreciating this way of measuring. Al, what do you have to add about this approach? Do you agree with Pat? Do you disagree?
I think that, obviously 100% agree with Pat. The complexity comes in areas of science where the answer is not quantifiable. And so for a protein structure, we have a statistical description of what the answer is, the structure, like at least a model of the structure that can be parameterized. For binding of small molecules to proteins, you can measure it with assays. Some of the newer methods where you're trying to model a cell, like what is a cell, right? A cell has different states, and how are you going to know if you're closer or further away? So personally, I think there's a need for thinking about benchmarking critical assessment in the new areas of biology where there is no ground truth, where we are trying to benchmark against a fuzzy answer. I think we can't ignore it because people are doing it, but it's the reality of what we're trying to accomplish today. So I personally think that the community, instead of starting 42 trillion different benchmarking projects, there's the brain trust in Pat and the Dream guys and Cash and CASP, who've been doing this for a long time, to come together and think about how to properly set up critical assessment of these fuzzy answers. It's relatively simple in small molecule binding, which is what we do. It's relatively simple in structural biology. It's incredibly complicated in these new areas. And I think the community will be disserved by thinking benchmarking is an easy thing to do and anyone can do it and everyone can run their own. I think there will be great value in coming together, getting the folks who really know what they're doing to help create a community of scholars interested in benchmarking. And I know that the Dream and the Cash and hopefully Pat's team are coming together in an organization they're going to call Beacon, which will do exactly that, which will serve as lessons of learned, provide guidance to the community, and sort of think about the new challenges in critical assessment. So I'm kind of scared what's going on now, where everybody's doing it, and it's so cool. It worked for AlphaFold, it'll work for everything, and I don't think that's true. The data aren't there, the methods aren't there, the simplicity is not there. And what we're going to get is we're going to get exaggerated claims of performance, right? Everybody thinks their method's best, and they'll find a benchmarking competition that shows that, and it's going to be a zoo. Okay, that was all my inside voice.
No, I really like the way you put it. The way I think about it, long ago, I was part of a team working on AI with chip generation at IBM. So we analyzed the fab, how the chip is created on a wafer. And this process has different steps, and there are engineers expert of each of the steps. So there's engineers who are expert of photolithography and engineers who are experts of etching or deposition, the different steps where you created the wafer. And at the end, what you want to do is a successful wafer that brings money and the chips works well. But each engineer has its own measurement for each of the steps. And the way I think about it with the ADME and the protein structure and so on, you each look at part of a process, but at the end, you want a drug that performs, that it's not only you understood the chemical properties, but it interacts with the proteins in a certain way, and it affects the cell in a certain way, and therefore it affects the body in a certain way. And I haven't seen yet, and maybe leaders like you can generate this kind of thing, I haven't seen yet someone who looks at the whole process of the production layer, right? You have all of these components on this system, And you want to have the end results. It's insufficient to have good results on each and every step, right?
I think the analogy falls. When you engage in a material science process, you can quantify whether the previous step worked with quite accuracy, probably with very strict parameters. Half the proteins in the human genome or genes in the human genome have fewer than 5 papers. We know, we can quantify, we are ignorant of human biology. So imagining that we can start to predict what happens when we put a molecule in a person is ludicrous, given the data gap that is quantifiable. So I just don't think it's going to be that easy.
Anytime soon.
No way. It's been 27 years since the human genome was sequenced, and literally there are 9,000 genes with fewer than 5 papers. So how in the world are we going to understand the system if we don't understand the functions of half of our genome? It just makes no sense to me. All these people trying to model biology and little cells and seeing things going up and down and saying.
I agree. But I think there's still tremendous value in being able to address pieces of this puzzle, because there are still a lot of parts of this that we're not very good at. So yes, I'd love to be able to model an organism, but I agree with Al. I think we're a long way from there. But I think, just in terms of looking at being able to predict how a small molecule binds, being able to predict its binding potency, being able to predict whether a particular drug is going to be metabolized. Even these small pieces can save us, months or even years in the drug discovery process.
And do you think these small pieces require tailoring between different benchmarks? Do you envision that you have a benchmark on ADME and a benchmark on a protein, and then a benchmark on the binding between them, and then a benchmark? So you need to create some consistency across the benchmarks themselves.
I think if you could get to the point, I don't know that the benchmarks need to be consistent, but I think that there's a lot to be learned. And, this is one of the things Al and I have talked about. If we can get to the point where we've got benchmark efforts and we're all looking at the same compounds, even, I think even there, then you can get to the point where you can start to integrate the data and start to look at things in a more multimodal sense.
I should say that the data collection, for those who aren't experimentalists, it is a real slog. The care with which you have to collect these expensive data, these aren't data you generate in an afternoon, right? These are $1 million experiments. The attention to detail, the quantifying the variance, the reproducibility of it, doing that right is super duper expensive. And that's why it's not been done before. And that's why, as you intimated and as Pat said, it has to be done by a large group working towards one cause, I think, because the resources are too large for someone just, I'm going to generate data in my lab. That's literally impossible.
That leads to the next question. So yes, there are these very large efforts, which is great. And then there are like students joining teams and asking themselves where they fit in, what would be a good place to start. And do you have a recommendation of, Tomer can represent the challenge himself. He just recently started PhD, right? And you ask yourself, you can say it better than I, maybe, Tomer, maybe you can express what you see from your perspective.
Yeah, so I think the most important, for the benchmark for a data set would be usability has to, in my opinion, again, if I'm look on other fields like images or speech, has to have training set, validation set, and test set, and the test set has to be not just randomly chosen like a lot of people are doing. It has to be actually different. And I guess the data set that people are using are the ones that are publicly available and are curated well and the test set and the metric used are really fitting for this data set.
So what you're saying is that in other areas, there's much better organization and information than what you see in this area, right?
For example, I can say like we did recently like paper for antibody design, the novel antibody design, and we find it was really challenging to compare ourselves to other methods because everybody uses a different test set and they trained on a different training set. So you don't know if you're better, your method is better because you trained on a different training set or because your tool is actually better. And then you want to compare, some people are doing sequence recovery, some are doing sequence identity. So it's like everyone can choose the metric that we're the best at and the test that we're the best at, and then see everyone is like the best for antibody design. And if you look at paper, this problem is like already solved, right? But we know it's not so. And also, again, we are looking on antibody design. We have this problem of like, again, as I said, most of the paper are doing like some sort of sequence recovery, or which I don't know if it actually means anything for like if it really means that the model is successful, because we don't want 100% sequence recovery, but we don't want 0. So yeah, without like really like independent tests, like as they said, like CASP or CAFRI or stuff like that, it's really hard to really tell apart the good models and the one that overfit on their training.
Thank you very much, Tomer, for putting this so nicely, these questions, maybe to Al and Pat, who have this huge experience with the community. What would be your guidance to students who look for more order and better ways to measure and so on? What would be your recommendation?
I can
go ahead, Pat.
I have very strong feelings on this. When I look at building a machine learning model, there's 3 components to it. There's the data, there's the representation, and there's the algorithm. The vast majority of academic work is focused on the algorithms, which is not what we need. We don't need another diffusion model. We really don't. And if you look at meetings like NeurIPS and ICLR, all the work is on algorithms. We need to focus on representation, especially as far as small molecules goes. We don't have good ways of representing small molecules in a machine learning model. We're using the same molecular representation we've been using for the last 40 years. We need to get beyond that. We still don't know how to integrate 3D into our models. We don't have models that are interpretable. I don't want to go to a drug discovery team and say, give me every molecule that you can think of, and I'll tell you which ones are good. What I want is a model that's interpretable, so I can go back to the team and say, no, you should change this part of the molecule, you should leave this part alone. So I think all of this comes from representation, and I think we need much more of a focus on molecular representation.
Thank you. Maybe one of the reasons you see this misalignment between the need and what the teams are working on is the incentive system. A PhD student wants to have a new algorithm that is highly referenced. Not always you receive, it has this aura around it. I had this amazing idea of how to do it. Representation doesn't have the same public relations.
I think there's also a problem with the way that we're training people. I think we need to train people to sit at the intersection of disciplines. I don't think you can just know computer science and develop a molecular representation. You have to have that understanding of chemistry and biology coupled with the understanding of computer science.
Yeah, I agree. I would start as a PhD student, choose an area of science like chemistry or biochemistry and go deep and learn all about the experiments and learn about the the deep science in the area that you want to make predictions. I mean, if you look at, David Baker, he was an outstanding, he understood proteins before he started to do this. The AlphaFold team, a lot of them were structural biologists and sequence analysis. I think the algorithm advancements are going to be incremental compared with the idea of how to think about the underlying science in these particular areas. LLM works because everyone knows how to read. You don't have to learn much to learn how to read, right? And so 100% agree. If I was actually training students, I'd say go get a degree in organic chemistry or go get a degree in biochemistry in our fields of drug discovery or protein structure for the protein world, not in algorithms or comp sci. That can learn later.
Thank you. These are very interesting things. And the question is also, okay, you have good comments and recommendations now to students how to prepare themselves to be strong contributors in these areas. How much do you see acceptance of the benchmarks? Do you see there are some challenges? How does the benchmarks contributing to trust among clinicians, researchers, regulators? Do you see that well accepted, or there are some hurdles in the way of benefiting from the benchmarks?
So on the clinical side, I'm not as familiar, but I see that with the increasing number of benchmarks, the ML community, which is, not small, but restricted in the number of things that they can participate in. I think their interests is being scattered too much, and you don't have enough thought about. So maybe one could use a consolidation of these benchmarks so that people could really give their attention to each and every one. Because if you participate in let's say, a radiology thing, you probably should understand what cancer is and think about the biology instead of just treating it as a static image, and understand all the different classes of tumors. For chemistry ones, you better know some chemistry. It takes some time for the machine learning community to come up to talk with folks in that field to understand how to make better predictions. And so that's how I see that with too many benchmarking organizations, there's going to be too few people competing. There'll be unclear answers, especially in areas where there is no quantifiable answer. So I think that if we're going to get the trust of the regulators and stuff, it has to be a much more, in my opinion, formalized benchmarking teams that set sort of standards on how one runs these things and measures these things. Data quality, you shouldn't run a benchmarking competition unless the data are beautiful, I think, or else it's just going to be modeling noise. Okay, that's it again. Over to Pat.
Yeah, I agree completely. I think these benchmarks serve as an important reality check for the field, because I think a lot of times people are thinking that this is all a solved problem, and then we run something like CASH, and it's like, hey, wait a minute, no one did well here. And I think it helps to point us in the right direction. I think sometimes, though, benchmarks are giving people the wrong impression. I think people saw the success of AlphaFold at CASP, and now, I talk to far too many people who think that protein structure prediction is a solved problem when it's not. So, I think so much of this really is coming down to how we communicate the results. And I think it's important for those of us who are running these challenges to make sure that we're not painting too rosy a picture. I think we can use this as a mirror for the field, and we can use this to figure out what's working, what's not, and where we need to focus. But let's not go out and say, great, solved problem.
Another thing that we were thinking about the need to sort of maybe even consolidate some of the benchmarking is each of the benchmarking organizations, everybody, has to raise their own money for it. And it's a time sink to have to spend your time, like John, you saw the news about the CASP, with the NIH troubles, it was delayed for 4 months, and that would have been cataclysmic. And so it takes time and effort to do this. And I think potentially, and also, you know, as folks retire, the learnings are gone. It'd be great to have sort of an organizational entity that could, you know, make it easier to raise funding for the benchmarking and also share experiences across the different types of challenges so we can do it better.
Yeah. So a related question. Sorry, Pat, you wanted to add something?
No, I just wanted to say I agree. These are very difficult to do, you know, and there's a few of us who've made all the mistakes over the last 25 years. But you know, this is a non-trivial thing to do, and I agree with Al. We need to get to the point where we can consolidate these learnings and make sure that we're not out there making the same mistakes again.
I completely agree, and I have one last question before we open it for if there's someone from the audience who wants to put in the chat question. You are welcome to do it already now, or when we finish with this question, you can just jump and raise your hand. One last question. You said there are so few of you who have all of this experience, and it's such a time-consuming effort, and the budget is limited, and the community is complicated, and so important to paint the right picture and to create the right communication. The question is, do you think something changed because we are in this era of Gen AI and foundation models and ChatGPT and the like? Does it mean it's easier? You're going to do the benchmarks in a different way?
To me, there's going to be a new method, this method, next year. The proof is in the pudding. Compete and see, and then we'll know if it's the right way to go. Yeah, one thing, Pat, sorry, I forgot to mention, you know, the dream challenges that have been around for 20 years, IBM should take credit for helping start those. I mean, it was a very interesting and important contribution to this field. Go ahead, Pat, sorry.
No, it's
thanks for the credit.
Yeah, I think it's nice that we've got, you know, perhaps the price of entry is less than it was in the past, and you know, a lot of this has become democratized. A lot of machine learning tools are out there for people to use. So, you know, my hope is that this will bring in a broader community, and especially bring in people more from the experimental side who understand the data, and perhaps, you know, get to a point where we can have more collaboration between people who understand the data as well as people who know how to build models.
Agree. And I see that some people commented in the, Heidi made some comment in the chat. Is there any question, last question before we close this amazing panel? Thank you very much for sharing your views. This was very fascinating. Anyone else wants to ask a question?
Yeah, Michal, actually, just a question. I was going to put it on the chat, but I'll just ask it away. Just because we have very concrete example in single cell analyses, but actually in cell type annotation, where we're seeing in new papers coming up with new benchmarks, right? And I've taken these are often created by the machine learning community, and I'm often, every time that I bring these to the biologists and to genomics experts, they don't agree with half of the way in which the benchmarks were created. So I just wonder if the panel, what can we do to make better benchmarks, or to make benchmarks that we can be trusted by the biologists and the experts in the field? Do we need to bring them in to the discussions earlier? I mean, how do we close this gap here between the technologists that are creating benchmarks and then the biologists? Thank you.
I would be cautious about painting us all as biologists. There are quantitative scientists who do biology. There are phenomenological scientists who do biology. This is what I think will require a think of all those, as I said, this beacon organization we're trying to put together to think exactly about that. Because those are, the single cell is a perfect case of where it's a fuzzy answer. And the readout is fuzzy. They usually do transcription analysis, which is only sees a fraction of the RNAs in the cell, as really sort of subject to when you harvest the cells, what cell culture conditions. There's just so many parameters that are difficult to control, so that even the data are so fuzzy and the answer is fuzzy. And then you're trying to benchmark fuzzy against fuzzy. I think this is not going to be a simple answer by getting guys in the room or people in the room. I think it's going to have to take that as well as some deep thought as to what is the proper way to approach a generic problem where the answer is not an answer.
You know, these cell competitions are based on experiments, so
noisy experiments that may not reflect reality. Yes, they are based on experiments. That's what I mean, right? It's a super interesting phenomenon. The kind of experiments that Pat and I do, we spend a lot of time making it very simple, so it's not grandiose. It's does this bind to that, and what's the affinity, or what's the shape? It sounds very small, as you said before, very focused, very niche, but there's an answer. And we know you're better or worse. The fuzziness of the cell enables everyone to declare victory because there is no answer. And then you'll have debate over whether it was the right model, debate over it's the right cell, and you'll get nowhere. You'll go round and round and round and round and get nowhere. So it might be a little harsh, but it's getting close to reality.
Pat, do you have the same views? Do you want to share your own view?
No, I agree completely. I think really the best way to put these benchmarks together is make sure that you're involving the data consumers in this. So you want to not only involve the people who are generating the data, but involve the people who are using the data and understand how they use the data, what their concerns are, and make sure that that's part of the generation of the data sets. This is one of the things that we did when we started putting together the Polaris effort, was we assembled a group of people from the pharmaceutical industry and from academia. And we really spent a lot of time as a group talking about what we considered to be appropriate for data sets. So I think it has to be a community effort.
Thank you very much. And I see people are agreeing with you also over the chat.
Yeah, Pat and I had to find something to disagree on. This is not fun.
In the area, there is a question from Raquel, if I pronounce right your name, Raquel. In the area of molecular representation, perhaps Pat could comment on the progress made in graphical representation for dealing with 3D small molecules information. Do you want to comment on that?
Sure, yeah. I think actually there's a fundamental machine learning issue here that people don't appreciate sometimes when it comes to using 3D structures of molecules to build machine learning models. I think we all appreciate the fact that small molecules are flexible and they exist in multiple conformations, but nobody really knows how to integrate that conformational flexibility into a machine learning model. If you think about what a machine learning model is typically designed to do, it's designed to take a single instance and relate it to a single label. I have a picture, that's a picture of a cat. That's not what we want to do with 3D structures of molecules. We want to take this bag of instances, right, this set of conformers, and relate that to a single label. So that's a field called multiple instance machine learning, which is very, very poorly developed. You know, there's probably a dozen papers out there on this right now.
Do I hear you asking people to develop new algorithms?
Well, I think it's a combination of the representation together with an algorithm there. So yeah, okay, I've contradicted myself already. But you know, I think we need to be able to come up with a way, whether it's a representation or an algorithm, of taking this information that comes from a flexible molecule and integrating it. You know, if you take a look at the molecular representations we currently use, they think that benzene and thiophene are very different, when as medicinal chemists, we know that they're bioisosteres.
Yeah, thank you very much. Maybe last question that we have from Canada. Would love to, so I guess it's for you, Al, but I'm not sure. Would love to hear thoughts on new protein language models like GPT-4b micro to design enhanced protein variants. Do you have any thoughts on that?
I don't actually pay much attention to the, I'm not going to say incremental, but to the various ways in which folks design stuff, because there's just so much stuff going on, and I'm not sufficiently knowledgeable enough to know whose little method, I didn't say that correctly, but which method in the competing world of methods. That's why benchmarking for someone like me who's a little bit distant from it is so perfect, right? If I can get a trusted entity to set up a nice benchmarking competition with metrics and a group of really experienced critical assessors, then I'll know on what these new language models do. Right now, it's silly to say I like this one and not that one, because everyone sounds good until they're measured against the same way. I'll never know. So for someone like me, the benchmarking is fantastic, because I don't have to be diving into every paper. Is this one? Is this one? I just let it all sort out by some fair adjudication.
It's interesting. I will end with one last question, because when you talk about how important the benchmarking is, I think it brings the question of trustworthy in the technology independent of the benchmark. Let's say a technology was outperforming. Do you need it to be somewhat transparent to understand how it works, or you don't care at all? You just want it to perform on the benchmark and then you would adapt? How important it is to be able to understand and learn about the technology itself?
Go ahead, Pat.
Yeah, I think it's essential. I think the purpose of these benchmarks is not just to allow someone to claim victory, right? The purpose of the benchmarks is to raise the level of the field. So what I'd like to do is, if someone does come up with an improvement, I'd like to know why, so that we can build upon that.
I think absolutely. It's also fair. The amount of time and effort experimentalists take in generating these data for the public good and willing to share them, it's a lot of zeros. And I think it's incumbent on the folks who are using those data to give back, as it were, and so that the field can advance. We're generating those data. We're in the laboratory, standing up, doing lots of experiments, going to the cold room to generate data for the public good. I think it's only fair that everyone else reciprocates.
Thanks a lot. That's a good note to end this discussion on fairness and contribution and community work. Thanks a lot, Pat and Tomer and Al, for sharing your thoughts. This was fascinating. And we move to the closing remarks by Yanis. Am I right, Joao?
Yes, we do. Yeah. Yanis, are you on the call?
One second, I have some. Can you hear me?
We can. Yes. Yeah.
Okay, fantastic. So I will not take a lot of time, just to thank everyone for coming together today. I learned quite a bit. It was a fantastic event. And I think also the panel discussion was very fascinating in terms of pointing out what may be some of the issues when people that have a lot of expertise in the algorithmic world, but they don't understand necessarily the domain, try to make contributions in an area where domain knowledge is quite important. Let me also mention, if you can move to the next slide, that this event was also co-sponsored by the AI Alliance. So the AI Alliance is now a global alliance that, as you can see in the slide, includes more than 195 organizations from 29 countries. And it's an alliance of organizations and commercial entities and universities that believe that open AI models would benefit everyone and want to contribute to this creation of these open AI models. The community that is behind the AI Alliance now amounts to more than 25,000. And there are many, many different projects, 30 core projects and more that people are working on. And I think there is one more slide. So the way that the Alliance works is that there are a number of different pillars and people can essentially join these different pillars. There are open agent labs where researchers, engineers, and subject matter experts can collaborate in creating open reference agents and components. There is an open data and models pillar where data sets, trusted data sets, and model development happens. And there are groups that discuss and debate standards and governance, addressing end-to-end challenges on deploying AI and AI agents at scale. And there are also other groups that are discussing policy and also coordinate on advocacy for educating the world, the AI world in general, and push for open innovation with public and with the public and with policymakers. So I think on that note, thank you for joining today. I should mention that this is the second installment of the workshop that we did. We had another workshop in person, a hybrid one, last year that took place in Boston at Boston University that we hosted here at BU. And we look forward to the third edition, perhaps at some point next year. Thank you for joining.
Thanks. Bye-bye.
Thank you all. Thanks.